A very good morning, everyone. Once again, we welcome you all for the second day session and wish that today's event leave an indelible mark in your memory. Welcoming is the noblest act of any function. By welcoming, we make the persons feel at home amidst us. May I request Dr. B. Harinathan, Assistant Professor of our department, to introduce today's man of honor. I welcome you, sir.
Good morning, everyone. I am really delighted to introduce today's resource person, Dr. C. Jayaprakash sir, to this international webinar on NUTRIGENOMICS. Dr. C. Jayaprakash sir has completed his post-graduation and doctorate in microbiology, who is also a qualified food analyst certified by FSSAI. Now he is currently serving as a lead auditor in food safety management system. He has a very good experience of nearly 24 years in R&D sector, and he has also helped to get accreditation of NABL accreditation in 60 national and international laboratories. His thrust research areas are food microbiology, food engineering, food processing, and etc. He has also developed 8 novel technologies for Indian Army. He has also filed nearly 11 national and international patents. He is also a proud recipient of 16 awards like Best Scientific Worker Award, Best Teacher Award, etc. etc. We are happy to have you in amidst the gathering, sir. Thank you. Sir,
Unmute yourself, sir. Your voice is not audible, sir.
Sorry. I'm audible?
Yes, sir.
Yeah. Good morning, one and all. Thank you for an invitation to give you a talk on this good seminar. Thank you for giving me this opportunity. It's really I have to thank because you have given the first session to me. So all are maybe very open to hear my voice and my words what I am going to speak today. So to my talk, it will be one minute. I will share the screen. Can you see my slides?
No, sir. Yes.
Not visible.
Yes, sir. Yes, now visible.
Visible. Okay. Thank you. So today I'm going to talk about some of the information about what is some small introduction about my organization. That's a DFRL, Defense Food Research Laboratory, Mysore. And what is Indian food industry? What are the opportunity as a microbiologist or biologist we have in front of us? So let us discuss about our Indian food industry status and whether we are having any opportunity in food processing industries, what are the new technique has come to the food processing, particularly for preservation of food for longer shelf life. Finally, I will conclude with my contribution. Particularly, I have developed some of the technology for food safety issue, microbiological food safety. So let me start with my talk by introducing my organization, that is a Defense Food Research Laboratory. It is a very prestigious organization. It's established in 1961 under Ministry of Defense, DRDO, Defense Research and Development Organization. So our main motto, our aim is to cater the needs of the food product for the three services, particularly Army, Navy and Air Force. So as you know, our service forces, they have to work in a different environment. They have to work, they have been placed in a different locations where it is highly different environment, particularly harsh environment. They have to work particularly in northern region. They have to work in a high mountain area, particularly very cold. So it is a very high. So they have to work in a very unfavorable environment. And Western, they have to work in, some people are working in a very desert area, some people work in submarine. So different location they are working. They have to be placed. Sometimes what will happen, they have to fight against the enemy, along with that they have to fight against the environment conditions also. That is a situation. So these type of service force people, when they are in the operation, what type of food we have to give? That is our motive of DFRL. We used to develop different type of technique and technology. Particularly whatever food which we are preparing in our home, that has to be converted into a very convenient, ready to use with nutritious, without any preservative, and it should have a longer shelf life. So this type of product we used to develop, and we will make the one day ration or combo ration. So different type of ration for this army troops with the different requirements and different location, what type of food we have to give. This type of work we are doing. So apart from our Army and Navy, Air Force, now recently we are working for the product development for our Gaganyaan mission. So our Indian Corps, in ISRO, they are planning to send our people to space. So we are also developing the food product for them, and we are also developing some of the items which they have to carry along with them. So these items we are now working, and we have developed some technology for even astronauts. So you can see here some of the important technology developed by DFRL. Particularly, we are the pioneer in retard pouch processing technology and self-eating system. When the food is going to in high altitude area where almost the environment condition is -30 or less than that, so food as such it will become ice. So how our army people, they can eat that ice? They cannot eat. So we have to provide some of the system which has to heat the product, and it should make warm condition to eat. So we have developed some of the self-eating system based on the exothermic reaction. So we have different type of food product. Apart from the retard pouch process, we have dehydrated foods for reducing the weight of the ration. We have freeze-dried dehydration, hurdle technology, minimally processed fruits and vegetables, and we have developed even BSL-3 mobile lab for tackle the food defense application. I will talk about more about the BSL-3 mobile lab for food defense application. And we also develop a portable microbiology laboratory. That also I will be discussing more about that. So we have, these are the only small I have brought. So these are the glimpse to you. So what area we are working. So you'll come to know. So we have developed whatever the food we develop here, that will be the part of our army ration. So one army ration will have breakfast, lunch and dinner. So all will have the ready to eat foods. Only warm it, or as such they can eat, they can heat and eat. Some of the food product they can, any emergency situation we can drop from the air, from the helicopter or any craft, so they can take it and they can use it. So apart from this type of technology, whatever we are developing the technology, we have some pilot plan. We can produce some of the emergency situation. We can produce some quantity of food and we can supply. And during Kargil war, we have supplied a huge number of food, ready-to-eat foods to the army Jawan. Particularly, it is all type of ready-to-eat foods became famous after the Kargil war. Whatever our technology came to limelight and because of the Kargil war times food supplies. So let us talk about what is interesting area in food. As a microbiologist or a biochemist or any other biologist, is there any future with the food sector? Yes, India is a very blessed country which is having very good resources and we have very good climatic conditions. So almost we can see almost world largest irrigation crop area we have. So we have two monsoons, so we can cultivate twice in a year. So two harvests we can make it in a year. So after the green revolution, we have multiplied our production, food production into 4 times, particularly in food grains 4 times, milk is 6 times, horticulture crops almost 6 times, so meat. And also we have a very long coastal area. We are getting very good fishery and marine product for export and our application, our usage. So the world population mostly, we ourselves, India is having almost 17.74, more than 17% world population itself we have. So we have big population and most of the people we are living and we are depending on, almost 60% of people depending on the agriculture. So everybody is maximum, they are working for going to the cities. So though the many people they are migrating from the villages. So there are a lot of scope in agriculture and food processing area. So we traveled from food shortage to now food security. We are able to produce huge quantity of food. We are having self-sufficient food now. And our Indian food sector is a main sector in our economy also. So you can see here our dairy sector. India is the largest producer of milk in the world. And India is the second largest producer of fruits and vegetables and second largest producer of meat, egg product, and third largest in broiler meat. So third largest in fish and exports also. So you can see lot of product we are in the number 1 and 2 or 3. So we have the chance become a food factory for the world. That's what one survey, McKinsey survey says, India can become a food factory for the world. So why not now? So we once is having lot of scope, we have to put some effort to make this to happen. So if you see our Indian agriculture, so we have now self-sufficient of primary production, and we have also huge demand, the consumer end. You can see the ring The first thing is the primary production, secondary, third one is consumer. So only weak section is food processing and storage. As per the ICAR data says almost 10% of our produce is getting wasted. How to overcome this? So we have to have a good processing and storage facility to keep our country continuously as a self-sufficient food security, nothing but a self-sufficient food for all. And at the same time, we should have the export, and the revenue has to be improved for the agriculture sector also. So in front of us, what is the challenge is to ensure that continuously every year the population is also going up. How to meet the challenges in nutritional security? We have to meet through not only primary production, we have to have the conversion from perishable to longer shelf life product. We have to convert whatever agriculture commodities are coming, so that has to be collected and it has to be converted into longer shelf life without any spoilage. So you can see now milk has have a very good example. So now because of the society, so milk is being collected and it is converted into a value added product and it is going to the market without much spoilage. So same way we have to work for all other perishable product. And food processing increase sometimes seasonable, that has to be converted for shelf life with more shelf life. And it can be even easy when we are putting food processing, it is easy for us to transport from one place to another place. So if you put more food processing area, so we can improve the rural income by value addition of any raw material, and we can save the transport cost by converting high volume product into low volume product. So we can create job leading to poverty reduction and rural urban migration can be stopped. So you have to have the weak link, that is a food processing area is having lot of scope to improve and bring lot of changes in the farmer's life also, and addressing the issue of unemployment. So why the processed food or food ready to eat foods is very important? So any food product as such, when it is fresh, it is growing at the field level, it may contaminate with a different type of microorganism. So it may spoil the product as early. So once you have processed, minimum processing if you are doing, so that can be eliminated and you can extend the shelf life.
Sir, excuse me, sir.
Yeah, madam.
So the slide is not changing.
Not changed.
Yes.
It's coming, madam, now?
No, sir. Yes, now it is moving, sir.
Moving.
Yes, sir. At the end, it has moved. Also, could you please make the slides to be viewed in full screen, sir?
Madam?
The slide need to be viewed full screen, sir.
Slide should be
in full screen view, sir.
It is not full screen view.
Yes, sir. During your presentation, it is not viewed in full screen.
Oh, sorry. Is presently visible?
You are presenting, sir, but the slides are not visible.
Not coming. Yeah. Here also it's not coming. Can I join again?
Yeah, sure, sir. Or else we can do one thing, sir. You better share your slide in our mails. Microbiology@smc.edu.in. I will keep it in the chat box. You send your presentation to this mail ID, sir. We will share your presentation and you can present it, sir.
Yeah. That I can do it.
Okay, sir. You just send your PowerPoint to this mail ID, sir.
Yeah. I'm sending, ma'am. I'm sending.
Okay, sir. Okay, sir. Okay,
So
sir.
you can present from there, yeah.
Yes, sir. Yes, sir.
It is 25 MB. It's more than 25 MB. That is the problem.
Sir, or else can you convert your PowerPoint as PDF, sir?
Yeah, PDF means is easy.
It will be much
The
better.
size is more. Yeah, size is
Okay,
more.
sir. Or else convert it as PDF and then once again you can present, sir.
Send, madam. Just to see, madam.
Have you sent the mail, sir?
Microbiology at
VHNSN.in. Yes, received, sir. It is received. Just now it is
received.
received.
Yeah.
Let me give few minutes, sir.
Yeah, you can. Maybe net may be slow. That is the reason it is not coming out.
Okay, sir. Let me take few minutes, sir. We will present
Fine.
it soon.
You can put it full screen.
Is it okay now, sir?
Full screen is not possible, ma'am.
No.
You are visible there. It is visible to you now.
Yes, sir. Yes, sir. It is visible to us.
Yeah. So it can be moved, madam.
We can just zoom the size, sir. Do you need to be zoomed? Need it to be zoomed?
Yeah. So it is enough. Okay, madam. You can go next slide.
Few minutes, sir. Sorry, sir. Sorry for the inconveniences.
Shall I try again one more time presentation? My presentation.
Now it will be okay, sir.
Yeah. Even I can present from my PDF, I hope, because its size is less.
Okay, sir.
Can you?
If it is okay, we can go ahead, sir. Otherwise, you can present yourself. Is it okay for you, sir? Or do you
One
need...
minute, madam. 1 minute,
Okay,
you
sir.
close
Okay.
it.
Okay.
Sorry, you can present, madam. I will continue with that. Time is going on. You can present, madam.
Okay, sir. Okay, sir.
Yes. Yeah, you can go to the next slide, madam. Next slide. Next. Next one. Yeah. Previous one. You are visible this there, na?
Yes, sir.
Others able to see? Anyone can respond?
Any participants?
Yes, sir. We can able to see.
Fine, good. Yeah. So there are a lot of scope. We have to make our food product into a non-perishable food product. So let me summarize what are the technology available in front of us to convert any food into shelf-stable or longer shelf-life product. So if you take any product in the market or any technology, so it is only in 4 technologies available in front of us. Basic technology. One is freezing, another one is dehydration, removal of moisture and bring down the water activity. Third one is thermal processing. Apart from thermal processing, some irradiation also is there. That is a non-thermal processing. Next slide. So here I have summarized what are the products available with the dehydrated product. You can see any powders, any staple foods, any rice or any wheat or anything, whatever the agriculture crops, so any dal, anything, we are just sun drying and we are keeping the product for more than a year. So now drying of this type of perishable product, particularly the horticulture product, can also be done using this dehydration technology. There are different type of technology in dehydration, particularly spray drying is there, fry drying, defat frying dryer is all there, and extrusion technologies are there. So using this technology, you can convert any perishable product into longer shelf life product. Particularly, we have to bring down the water activity. Water activity, nothing but you are removing the moisture so that the moisture content is bringing down under the less than 14%. So the product is having less than 14% of moisture. So water activity will not be there. So microorganism growth will be controlled. So another technology is freeze frozen product. This is also very highly used in India. Also now, big plant they are putting for unfrozen. So they are placing the industry in the agriculture field itself. They are bringing all the agriculture commodity, particularly all vegetables, green peas, carrot, even all type of vegetables that can be converted into a frozen and you can be stored for more than even 3-4 years. You can store it under the frozen condition, particularly -40, and can be stored at -20 for longer shelf life. Then another technology is thermal processing. So thermal processing is occupying most of the ready-to-eat food product. Here one advantage, you can convert any perishable product, any food product, whatever we prepare in home, that can also be preserved by thermal processing and can be keep the shelf life more than 2 years, 1 to 2 years. So that is the technology, is very important technology where it's a ready to eat form we can convert and it is having more shelf life. So these are the main technology available in front of us. So basic technology will be same. If somebody is working on the R&D to make this technology, basic technology in improved form, that is a in future which we can expect. So what are the improvement in particularly in thermal processing is happening? So what was the basic, what is improvements is happening in thermal processing, that I will tell you in the next slide. Next slide please. Next slide. Yeah. So you can see now, previously this mango season, lot of mangoes will be come to the market and it was very cheaper rate and sometimes it is going on spoilage. Now so many company, because of this mango juices and other things, they have come up with a technology. There is a ultra heat treatment product just to make a pulp, and pulp can be preserved passing through the ultra heat treatment and fill in the aseptic condition in 200 kg weight in the drum. So that can be stored for more than a year. So this type of technology in India, it is coming and they have put lot of plant in wherever the mango cultivation is happening. So same way, if you go to even Erode one company, they have come up with one UHT plant. This is for only liquid product, food product. You have to convert the product into liquid and pass through the UHT heat exchanger at a 140 degrees centigrade for only heating at 4 seconds and cooling it and filling in aseptic condition. Here the product will be sterilized, the pouch will be sterilized, and the environment also is controlled aseptic condition, so it will be filled. So three angle, so the product is totally free from microorganism, so it will not get spoilage. Most of the food product is spoiling because of the microorganism. Chemical changes is very less, that can be addressed by using proper packaging. So the UHT products, even as I told near Erode, coconut, almost 1 lakh coconut they are breaking and they are extracting the coconut milk, and they are coconut milk. They are packing in 200-liter pouches, aseptic packing, and put it in the drum. So that is going to export. So this type of UHT process is very good for the liquid product to convert any perishable into longer shelf life product. So it is a very important for only liquid product. So solid product is having some constraint because we cannot pass through, we cannot pump it through any heat exchanger. So that is having different technology. So next slide. Next slide. Yeah. So next is thermal processing of any ready-to-eat foods, whatever we are preparing in home. So how we can convert this product into longer shelf life? Maybe vegetarian, non-vegetarian, like vegetable pulao or even rice or sambar or any other product. Maybe chicken curry or mutton curry, any product we can convert because it is having solid particle. It's a heterogeneous product. We cannot pump through the heat exchanger. So there is a one processing we have to do. But the retard pouch processing. Actually, what is a retard pouch processing? It is a combination of packaging and product and processing. So we have to make a product in a half-cooked condition, and we have to fill in a proper packaging, particularly retard pouch. It is made up of some 4-layer pouch. So it will be filled and sealed, and it will be arranged inside the retard. Retard nothing but a modified version of our autoclave. So we have to arrange inside, and we will give the heat treatment where whatever the half-cooked product will get fully cooked, and finally it will be eliminate all type of spoilage and other pathogenic microorganism. So that will be standardized by using putting the temperature probe, and it will be calculating the sterilization value, and finally we will be cooling and it will be taken out, and that product will be the shelf life of more than year. So this product, by using this product, so many companies now they are exporting their product, particularly MTR, ITC, so many companies are there. They taken this is a technology pioneer. DFL is a pioneer for this technology. We developed this technology by packaging also we develop, and the report that processing machinery and product also we have standardized. So many people taken our technology. Now it is in the market, particularly this just to take it, heat and eat. So all type of curries and other things are available in the market. So it is having a shelf life of more than 1 year. So some company they are exporting even one container of North Indian dishes like dal makhani to US every day, one container. So this type of exports also happening through this technology. So this technology is very economic way of preservation and processing of all type of perishable product. Next slide, please. This is what the picture I have shown here. So we will be placing a modified version of one autoclave, but here the temperature will be controlled, steam line will be there, the pressure control will be there by using the counter pressure. When you heat any food pouches, inside the food pouches, because of the heat, the pressure will happen. So we have to counter pressure using the air. So air will be there. So this type of some cooling will be happening in the high pressure cooling will happen. So this facility will be in this type of retard. So this product advantage is it is a commercially sterile nothing but it will not spoil at room temperature for more than 12 months. So no refrigeration is required. Ready to eat farm. So it is free from all type of chemical preservatives. So it's energy saving and increased quality attribute. Now if you want to improve the quality of the product also is possible by using this. So it is reduce the transport cost and storage cost. So DFL has developed a different type of technology, the product and technology for this retard pouch foods. So we have transfer the technology to more than 85 companies. You can see here MTR, ITC, Aldiram. So many people, they've taken our technology and they are exporting, they are having the market in India also. During this COVID period, this product demand was very high. So even MTR, their demand, they are unable to meet. So that much now demand is there. Even any software engineer, those who are traveling around the country, so they are carrying this product. So they need not to search the restaurant where Indian foods are available. So they can have at any country. This product only heat and eat. So this product became a very successful product because this technology developed by DFRL. This product is the part of our army ration. So this technology is very successful. Next. Next slide, please. So how we will process this thermal process? So when we take any food product or any preservation, we will keep in three things in our mind. First one is it should ensure the safety. It should be eliminate all type of pathogenic microorganism. Second one is it should eliminate the spoilage microorganism. By heat, we have to eliminate the spoilage microorganism. Once we are aiming for safety and spoilage, we should not lose the quality of the product. So we will be optimized based on that. So the product will be not under processed or over processed. So nutritional quality will be intact. So that way we will process. Particularly, what are the organisms we will take for ensuring the safety? That is Clostridium botulinum we will take. If you heat at 121 degrees centigrade for 4 minutes, 2.4 minutes, that is nothing but 3 minutes, that is ensuring the safety. If you are ensuring the spoilage, you have to take the organism for, it's the indicator organism Clostridium sporogens, where you have to process 5 minutes at 121 degree to reduce the 5 log reduction of this organism. So using these basic things, we will be optimize the product processing time. Next. So there are different type of retards are available for processing this. Steamer retard, water immersion retard, water spray retard. Now it depends upon the product and what are the packaging material you are using. You can select a type of retard for processing of this type of product. Particularly any product which is having only canned product, steamer retard will be very faster. So other small containers of any juices, other things and all, we can use even water spray retard. With different type of retards available with the different applications. The best one is water spray retard. You can reduce the heat process time when compared to other processing. Next. Now, I have already told that is a UHT, another one is retard processing. So UHT is very successful because we are heating the product only 4 seconds. But in the case of retard, we are processing for long time, particularly 121 degrees centigrade for 5 minutes. It is not possible any food product at 250 gram of food, bringing the product into 121 degrees centigrade is not easy. So it has to heat it from 30 to 40, 50, 60. It has to rise gradually and it has to reach 121 degree. From once it is reaching 121 degree, then 5 minutes you have to cool. So the total process time, if you calculate, it will come around 20 to 25 minutes you have to process to reach that sterilization value. So how fast you are heating, so that much you can retain the nutritional quality and texture and taste. So what is the innovation in thermal processing? So now people, mostly the frozen foods is having very good quality because there is no temperature adjustment. So all product quality will be intact when you freeze it. So it will be as natural as such. But it's a very, no, it is not the economic way. You have to have a cold chain on the place. So you cannot keep it at the room temperature. That is a constraint. So alternatively, this thermal processing. So thermal processing is, juices are very good. There is no problem because we are not heating more. When we take it a heterogeneous product, it is having some problem. It is taking long time to heat. Now one technology has come. That is nothing but MAT technology. MAT nothing but microwave assisted thermal processing. So these are technology now everybody is speaking. So the heterogeneous product, if it's chicken curry or mutton curry or any vegetable biryani, if I want to process, because inside so much of particles are there, the core area has to be sterilized. So you have to process for a long time, almost 30 to 40 minutes at 121 degree. So to avoid that process time, so the MAT technology has come. Using this MAT technology, you can reduce the temperature adjustment on the product. So you can increase the quality of the product. So that way, this technology is very much useful. Let me talk about some principle. Next slide, please. Microwave assisted thermal processing, nothing but here, when you apply temperature one side, another side microwave, so it is parallelly when you are giving into the product. So what will happen? This microwave always it will start the core area. So the core area it will be heated up. So from the outer side, the other steam can be applied and it can be heated up parallelly. So both things will happen parallel at a time. So the product process time will be reduced. When the come up time, normally we will tell that come up time. Come up time nothing but when the retard is bringing from 35 to 121 degree. That is bringing the product retard temperature from 35 to 121 degrees come up time. From 121 degree to holding time. 121 you are holding for some 5 to 10 minutes, then you are going for cooling. These are only thermal processing cycle. In this come up time, it is taking long time in normal thermal processing. When you put microwave assisted thermal processing, the come up time almost reduced. If the come up time is 20 minutes, means it is only 5 minutes we could be able to reach the 121 degree by using the microwave assisted thermal processing. So this technology is using, they developed in Washington University. It is for US Army they have developed, and it is also approved by the US FDA, this technology. So this technology now in India also many people they are looking and working. DFR also now our next program is develop a microwave assisted thermal processing technology. Next slide. So this is the way microwave which will go and now make excitation in the water molecule. So the water molecule will get pulled and packed for more than 2.5 billion times per second. So that will create the heat inside the product. Next slide, next slide. You can see here, it's a conventional heating. The heating is happening from outside. So microwave heating is happening from the core area. So when you put together, so the heating will be happening simultaneously and the heat come up time will be reduced. So the product quality will be reduced. Instead of 20-30 minutes process time, in the holding time, we are going to reduce by only 5 to 10 minutes, so that the quality of the product will be totally improved. It is only by cooking how much quality we are getting, same cooking effect only we are going to achieve by using the microwave assisted thermal processing. Next slide, next slide. So these are machines, the microwave machines is having preheating, microwave holding, finally it is going for cooling. So any type of product can be processed using this microwave heating, microwave assisted thermal processing. Next slide. So these are the product you can see here. These are the vegetable curry processed by microwave and normal thermal processing. What is the difference? You can see the color and texture. So the graph shows That is how much time it's taking to process by conventional method, traditional method and microwave method. It's a very short period. So how UHD is very beneficial. Same way microwave assisted thermal processing, it is having a lot of benefit to improve the quality of the product. It will not affect the nutritional quality product. Next slide. So some of the other non-thermal processing, as the heat is having thermal some effect on the nutrient, color and texture, we don't use other non-thermal processing. There are some technologies coming up, nothing but the PE of pulsed electrical field processing. So if you give some pulse electric field more than 20 kilowatt, so microorganism cell wall will be ruptured and it will go for killing, it will die. So these type of things also will have possible in any pumpable product, particularly any fluid product. So this technology is in foreign, they want to reduce any pathogens, they are using this as a pasteurization effect. They are using this. Next, another non-thermal processing, nothing but it is a high pressure processing. Using high pressure more than 600 megapascal for 20 to 10 to 20 seconds. This also is eliminating some of the spoilage and pathogenic microorganism. But these two technology cannot be used as a commercially sterile product because it cannot kill any spore forming bacteria. Particularly the spore forming bacteria, nothing but Bacillus or Clostridium, cannot be killed by this type of process. But if I want to aim to eliminate some of the pathogens like Salmonella or E. coli something, you can usually use this type of process. But this high pressure processing, you can use even solid product also. But pulse electric processing is only for liquid product. Next slide. So I am taking more time. Here I want to tell some of the innovations which I have made to our army applications. As you maybe all are a microbiologist, okay, if I want to set up a microbiology laboratory, you may think that large type of building is required, so much of equipment is required, so many workflow will be there. So I have made an innovation that any microbiology analysis is having 4 steps: sample preparation, sample inoculation, incubation, and interpretation. So these are the 4 steps in any microbiology analysis. How we can make this microbiology analysis simple? That way I have developed a portable microbiology laboratory. You can see this picture is a portable microbiology laboratory. Any microbiology analysis has to happen in a aseptic condition. Because once you are checking any microbial quality of any food product, there should not be any environment contamination. So how to avoid that? So I have developed a portable laminar airflow along with the incubator. Okay. Then I have developed a self-contained self-filter stomacher bag. Self-contained self-filter stomacher bag, where you can see here all-in bag. So it is a ready-made. So when we want to make any homogenization, first step is sample preparation. In homogenization, what we will do, we have to prepare your diluent, you have to sterilize, and you have to take to the sample preparation bag where you have to put your food and you have to put your sterilized diluent and you have to keep it under the stomacher. Stomacher nothing but the homogenizer. So it will homogenate the product and you have to take the product. You have to take the homogeneated sample for the further analysis. So this I have simplified. What I have done, I have made all diluent into a stomacher bag, which is having one filter also. So it is a sterilized 1 year shelf life. Just to put your food sample and you can crush by your hand also. It will be mixed properly. The filtrate will come. The sample filtrate will come other side. Just to take a known quantity 1 number for serial dilution, or you can take for the further analysis. So sample can be easily prepared by using this all-in bag. This is a very innovation. World, this is the first time I have made it. Okay, some other bags are available. It is not self-contained. So this is a self-contained, self-filter stomacher bag. Anywhere anybody can use it. Just to take the sample, put it inside, press it, the filtrate will come. From the filtrate, you can use the any Petri film. You know Petri film. Normally, when you want to go for the plating, you will be preparing the agar and you will sterilize the plates and you have to put your sample and you have to pour it. So many things are there. So now ready-made plates are available. That's called Petri film. Okay, it's a ready-made plate. It is already, the media is coated. Only 1 ml of this filtrate sample, you have to add it and press it, keep it in the incubator. This is like one paper, one small film type. So that can be put it inside and next day you can count the colony based on the you can process. And online.
Sir, your voice is breaking, sir.
Can you hear me now, madam?
Yes, now it's clear, sir.
Hello?
I can
Not
hear
here.
you now. Yes, sir, I can hear
Yeah,
you.
yeah. So Petri film is available. So many competitors have come. Now compact tray is also available. Instead of Petri film, compact tray also you can use it. So another innovation. So now with total plate count, I can do it by using Petri film. Sample, I can use it by all-in bag, which I have developed. So if I want to check it, this food sample, this water sample is having any coliform, E. coli or salmonella. So coliform, E. coli are very considered as a hygiene indicator of any food industry. So you have to test it by the standard method. It's taking more than 48 hours. So how fast you are able to test it, that is your challenge. So I have developed a single test kit media, which you can see coli E. I have given the name coli E because I can check it using this media, coliform and E. coli. So what you have to do, from the food sample, 10 gram of food sample, homogenate it and take 20 ml of the homogenate, put it into the coli E bottles, mix it. So initially the color will be purple color. Then finally it will, if it is any coliform is there, it will grow. It will grow and it will utilize the lactose which is present. So it will produce the acid. So whatever the lactose utilizing organism nothing but coliform. So it will become a yellow color. So acid production. Okay. So once you add some coax reagent in the top, okay, so you will get the pink ring. That is nothing but indole positive reaction. So lactose utilization, indole positive, gas inside one, Durgam's tubes also is there. It will gas production. So that will 100% confirm that single tube you are going to find out coliform, E. coli from food sample. If I want to put water, you can put water and you can incubate or keep it in the room temperature. You can count, you can see that your water is safe from coliform. Coliform, once the E. coli or coliforms are more, it is nothing but any fecal contaminations are happening. So by this test kit, it's one of the part of our portable microbiology lab. So total plate count, you can do it by Petri film. You can do using coli E to test coliform and E. coli. Another one more test kit, that is number Sal Plus, whether salmonella is there or not. This media is developed a specific media which only allow the salmonella to grow here. Any other organism will not grow. So we have prepared and using some antibiotic and some other technology. So other organism will not grow here. only salmonella will grow. Once the salmonella grow, so it will turn the media into black color. So this is nothing but hydrogen sulfide production. So it is, so once it just you put a sample, mix it and keep it. So it's a turn to black color. So you can 100% confirm that it is having salmonella. So normally any salmonella test, it will take almost 3 to 4 days. Pre-enrichment, enrichment, select your herd, then you have to go for the biochemical test, then finally you have to confirm with the serological test. So by using this test kit, you can test salmonella very easily. Yeah, next time. Yeah, there is a food safety portable microbiology lab. Now I have developed a mobile BSL-3 containment laboratory. Normally, any food processing unit, when you are exporting or anything, there is lot of threat is there. So if it is naturally any microorganism is contaminating, that is coming under the food safety. If somebody they are putting intentionally putting some type of pathogens inside the food material, let it go and contaminate the people and let the casualty will happen. So this type of intentionally, when you are putting any food, any microorganism or any type of chemicals, that is nothing but food defense. So how to overcome this type of intentional contamination of food defense? Particularly when you are suspecting any food defense, what do you have to have? You should safeguard yourself. You have to find out what is the inside, what is inside the food, what is they have intentionally contaminated. So we should have the laboratory which is having the facility of BSL-3 level. It should safeguard the microbiologists, those who are working inside. So we have made one 20-feet container into a mobile containment laboratory, mobile lab, BSL-3 lab. It is a negative pressure. You can see inside. It is a totally initially there is a 2 airlock we have given. So initially the sample will be given through side. That will directly go to the BSL-3 biosafety cabinet. So it will be taken inside and it will be processed and it will come to the decontamination room. This is utility. It is totally self-contained. It is having power. Particularly 15 kVA generator is there. It is having AC. It is having negative pressure, air handling unit, class 100. Particularly in biosafety cabinets are there. One PCR hoods is there. All type of molecular assay you can do it inside. PCR machines are there. So it has been placed in the Ashok Island chassis. You can carry anywhere and you can test any type of analysis from the food and biological sample. During the COVID situation, we were not aware that virus is how it is spreading. So there was a lot of problem was there. Then we given this mobile lab to the Mysore Medical College, and they were testing inside this lab very safe and very happily they worked, and every day they were doing more than 1,000 samples. 200 samples at a 8 hours period. So that facility we have provided inside. So the mobile BSL-3 lab, first time in country, you can carry anywhere and you can do the analysis at a customer site. So these are one of the contributions for food defense. Next slide. So I like to conclude because I have taken more time. So I like to conclude my talk. So R&D effort will really help us to extend the shelf life of any food product. All type of our traditional food has to be converted to a ready-to-eat form. Okay, there is a lot of scope in food processing area. Many engineers are coming inside to set up their laboratory, set up food processing unit. Okay, they are starting a lot of industries. So as a microbiologist, I am a microbiologist. After 25 years, I am having the experience. Now even any food technology cannot give having whatever knowledge. I have the same knowledge. So anybody can easily learn because the food is part of our life. You have to put your effort to simple learning. You can easily do any microbiology because the food technology nothing but is a 50% is microbiology component. If you address the microbiology, your food will be safe. So microbiology, those who are having the knowledge, they can enter into the food sector. They can be in the product development. They can be in the quality control. They can be the food safety management system like implementation of HACCP, good hygiene practices. So a lot of scope in food processing unit. So you can look for any good opportunity in food processing sector. Thank you so much for your opportunity. I hope you got some information. Thank you.
Really, we got lot of information, sir. Thank you very much for your effective presentation. It's our privilege to thank you, and we are very happy to have you as our guest of honor today. Thank you very much, sir.
Thank you. Thank you, madam. Thank you.
Now the session is open to you. The audience, if you have any doubts, any clarifications, if you want, you can ask him directly. So there's a question in the chat box, sir.
Yeah, madam, please tell me.
How much time will take to get the result for Coli E? Is it a rapid method compared to traditional VRBA plating method?
Madam, here almost 12 hours you will get the result. Maximum 12 hours you will get the result. Normally, other method it will take more time, and again you have to go for the confirmation. So that will take another 24 hours. So it will be 48 hours. So here you are going to get within 12 hours whether the coliform is there or E. coli is there. Easily you can find out. So this I have taken this portable microbiology laboratory. I have demonstrated in our army unit. Normally our army unit, they will procure lot of food items to supply to the unit. So in the procurement depot, I have placed our portable microbiology laboratory along with this all test kit. They are using regularly, and I am getting very good feedback from our end user.
Thank you very much, sir.
Any student, any questions? When we study, we don't have this type of opportunity to interact with any outsiders. I have seen many experts are coming in the line, so you are privileged to have this type of seminar. I hope you have to use it, and you can take numbers anytime. You can contact me also. Any carrier or any type of support from my side, I'm happy to give. Yes, I used to, whatever, I'm not a good speaker, but whatever I am innovative worker. So whatever we do, it should be useful for somebody. So that is the way I used to think, and I have to be, helpful for many students also. So I used to train the people. I used to send the people to the different industries, food testing laboratories, the things. If at all any microbiology, after if you like to enter into food sector, you can take my number. You can contact, or any help I am ready to give.
Really, we are blessed to, sir. Thank you very much.
Thank you, madam. Thank you. Thank you so much.
Now let us move on to our next session. If your action inspire others to dream more, learn more, and become more, you are a leader. says John Quincy Adams. At this juncture, I would like to attribute this proverb to our chief guest because their valuable past makes them important today. Now, it will be my honor to introduce our chief guest, Dr. Shugapriya Dhanasekaran, who is serving as an associate professor in Department of Medical Lab Sciences, College of Applied Medical Sciences, Prince Satam bin Abdulaziz University, Saudi Arabia. She has nearly 20 years of teaching experience. She is well expertised in molecular techniques, tissue culture, fluorescence, and confocal microscopy. She is a recipient of University Research Excellence Award, ISSCR Travel Award, DST Travel Award, Indian National Science Academy Award, Center for International Cooperation in Science Award, Young Scientist Award, etc. She is an editorial team member in various journals like European Journal of Preventive Medicine, Global Journal of Vaccine and Immunology, Virology and Immunology Journal, International Journal of Immunotherapy and Cancer Research, International Journal of Herbomedicine. She has published so many research papers in various national and international journals and published several book chapters. She has guided various research projects, and she is very much interested in investigating novel, alternative, safe, and efficacious strategies for treatment of cancer. And my words won't be enough in explaining her efficiency. We are privileged to have you, ma'am, and now the session is over to you, ma'am. Ma'am, please unmute yourself, ma'am. Thank you, ma'am.
Thank you, Dr. Vijay Lalita, for giving a wonderful introduction. First of all, I have to thank to providing me a wonderful occasions to Dr. Palaniyappan, the coordinator of this program, and Dr. Sundara Pandian, the principal of VHNSN College, and Dr. Prithvi Kumaran, the DBT coordinator, and especially Dr. Balasubramaniam, who invited me for this occasion. And my heartful thanks to my friend, my classmate, my research collaborator, Dr. M. Kannan. He suggested me for this special occasion. I am more happy to present my session. And so, are you able to see my slides?
Yes, ma'am.
Okay.
Can you please keep it in full screen, ma'am?
Yes, sure. Is it okay?
Yes, ma'am. Yes, ma'am. Now it's okay.
Okay.
Thank you.
Today we are going to discuss about nutrigenomics, epigenetics, and cancer preventions. So the human genome is comprised of approximately 3 billion base pair of DNA. The epigenetic modification, it can perform a significant role in various diseases, mostly for the pathogenesis and occurrence of the diseases. The DNA methylation, DNA acetylation, DNA phosphorylation, and ubiquitination are the major impact in the chromatin remodeling and most common in the epigenetic mechanisms. The most common thing is DNA methylation. Among the phosphorylation, acetylation, and ubiquitination, the methylation is the most common in the epigenetic mechanism. So many people mistakenly assume that the presence of certain genes or the single nucleotide polymorphism that may be the designs to the experience which can associated with the diseases. However, the most genes are flexible expressions and complex interaction with among the multiple genes. So that can affect the gene expressions. So these interactions we can known as the epigenetic influences. That epigenetic influences may involve with environment, diet, the chemical exposure, the stress, and other presence of some single nucleotide polymorphism. For example, some people's the same proteins may be coated, but the enzyme amino acids may be, instead of valine, methionine may be presented in the proteins that may be interact. So we will see what are this thing. Okay, a special diagnostic panel which assigns a functional nutritional status along with genetic single nucleotide polymorphism such as methylene hydrofluoride transferase reductase enzyme and catechol O methyltransferase, apolipin E, and tumor necrosis factor alpha, and other many genes are involved. So I just explain only a few genes which is involved in the nutrition with genetics. Okay, so with the aid we can have a personalized diet, lifestyle, and supplementation recommendation to the positively impact the physical and biochemical functions. So this presentation will focus on understanding of genetic predisposition and personalized nutrition which is necessary for our body. The nutrigenomics is a relatively a fresh discipline, but it possesses enormous potential than it can apply for prevention and management of carcinomas, cardiovascular disease, neurodegenerative disease, and metabolic disorder. Okay, so we can have develop a general understanding about nutrigenomics. We can have a combine of basic genetics with nutrition. How we can apply the diet and lifestyles which can prevent the disease like us diabetes, like a metabolic disease, and cancer. Okay, so we can learn about a special lab testing for this specific single nucleotide polymorphisms which can influence us on health conditions of individuals and susceptibility of the disease by defending the metabolic syndromes and gene expressions. So we can think that your genes are not your destiny. Okay, the food send the information signals to the gene, and the genes critically to determine the functions. The nutritions modify the extent to which the different genes are exposed, and they can modulate whether individual can have a potential genetic background or not, and according to that the genetic mechanisms may be differ. Okay, so the personalized nutrition is the important in the healthcare. So targeted dietary prescriptions as well as the individual based genetics and lifestyle may be involved in the disease mechanisms. So here there is a flow chart which is going to explain the functional medicines based on the health sector. Okay, so the genetics, which is the genotyping and single nucleotide polymorphism, both involved in the epigenetics like methylation and phosphorylation. Either sometimes methylation may be taken place or sometimes the phosphorylation may be taken place in the genomic sequence. So that resulted in the changes in the functional biomarkers. The biomarkers may be involved in the inflammatory markers or pro-inflammatory markers or anti-inflammatory markers or anything, maybe a cancer causing agents, or it may be different. So we have to analyze the epigenetic mechanisms. Before that, we have to analyze the SNP, then epigenetic mechanisms, then go for the functional biomarkers. According to that, we can have a healthy lifestyle. So here there is a correlations, connection, interactions between the diet and the genome. Food, nutrition, and genome, there is a big interactions. The nutrigenomics is a gene expression that affects the diet of the gene expression, and nutriogenetics, it's a gene variation, the individual the effect of individuals genetic variations to the diet. So there is a difference between nutrigenetics and nutriogenomics, but there is a interactions, interlink, correlations between nutriogenomics and nutriogenetics. We can modulate the genomics with our good nutrition. Okay, so the nutrigenomics is the influence of food to the genetic expression and how what you eat, which can turns on your genes or off your genes, maybe on of your oncogenes or off of your tumor suppressor genes. Okay, it can refers to the interaction between the genes and nutritions. It can modulate the modulations can occur directly or indirectly, like onset of the chronic diseases, incidence of the chronic diseases, or progression, severity, that mostly influenced by diet regulated genes and their common variants. Okay, for example, this is a broccoli which has a bioactive component, sulforaphane, which can turn off the oncogenes. So what is oncogenes? The oncogenes is a cancer initiating or cancer causing agents which can be off. Okay, so if continuous intake of this broccoli, it can reduce the cancer causing agents, okay, genes, especially cancer causing genes. Okay, and another one example is resveratrol. This resveratrol is mostly presented in grape skin and grape seed. Okay, it can lead to the changes in the gene expression and can shift the energy production and metabolism of the cell cycle. So it is the major thing and helpful for the cell cycle cassette. Okay, so why the nutrigenomic is more important for the nutrition? It allows you a central dogma, and we can think food is more than the calories. Okay, the calories is a calorie, but the bad food gives you a disease unless you have a genes to invent to protect you. This is we have to always keep in our mind. The bad food gives you a disease if we don't have a genes to invent to protect you. Okay, so the food is the full informational signals to our gene. It's the calorie is to be the judge upon the context which comes off. Okay, so we are continuously interacting with dietary signals which certain food enhances a beneficial nutritional and negative effect of the genes. So the clinical trials need to include the genetic variability and single nucleotide polymorphism as a major factor, so it can influence in the nutrigenomics. Then genetics makeup of a person can lead to require certain nutrition, may be higher or lower of the nutritions. Both it may be implicated in propensity towards the disease. So this can include the single nucleotide polymorphism as well as a play role in the personalized nutrition. So we have to go through that epigenetics and nutrition. Where is the link with the nutrition on epigenetics? There is certain bioactive food ingredients and micronutrients which are normally involved in DNA methylation, DNA phosphorylation, DNA acetylation. Okay, so the gene expression and histone modification mostly based on the DNA methylation and acetylation. So this involved in the biological and metabolic regulatory pathways. So the deficiency of any key nutrients, okay, like vitamins, mostly vitamin B, B12, B6, this is the most important, B6, most important vitamins. Okay, there is any deficiency in the key nutrients and absence of methyl donors in the inheritance of that methyl transferases, this can lead to mutation in a gene by activating the promoter genes. Okay, it may be activate hypomethylation or hypermethylation. This results in suppression of tumor suppressor gene, okay, or silencing of tumor suppressor gene. We have two type of genes. One is oncogenes, another one is tumor suppressor genes. The tumor suppressor genes always suppress the tumor genes. Okay, the oncogenes is the cancer causing agents. This genes always in sleep mode. So when we are taking, if there is any mutation taken place in the DNA methylation, hypomethylation or hypermethylation, it may be silence the tumor suppressor gene and activates the oncogenes. Okay, so that may lead to continuous proliferation, uncontrolled cell proliferation of the cell. That leads to tumor or a cancer development. So here I gave an overall diagrammatic representation that a genomics involved in epigenetics, metabolomics, and transcriptomics with proteomics. All over, it is the major control. See, for example, if you are intaking of a dietary mutagen, like some fast foods, example, the fast foods. So it which contains the dietary mutagens. When the dietary mutagens enters into our body, it methylates the DNA molecules. So once the DNA methylation happen place, activates the oncogenes. Activation of oncogenes resulted in cell cycle inhibition, cell proliferation and arrest of cell cycle inhibition. Okay, so the resulting in cancer. Okay, this is the major impact of the dietary mutagens, especially the fast food. Okay, so here there is one more examples. This is a arachnoidic acid which is presented in shellfish and red meat, fat of the red meat. Okay, this activates the COX-2 overproduction and resulting in eicosanoids. Okay, this eicosanoids is a protein which enhance which enhance the this is the cytokines which enhance the pro-inflammatory markers and cause the disease-causing diseases. Okay, it is a disease-causing. The eicosanoid is a disease-causing agents. Now, if we consume the omega-3 fatty acid or omega-6 fatty acid, that suppress the COX-2 expression. The suppression of COX-2 expression, which enhance the production of a different form of eicosanoid. Okay, this different form of eicosanoid act as a anti-inflammatory markers and suppress the inflammatory markers. So we can now decide which type of food is good, which type of food is bad for our health. Okay, so we can say that epigenetics is a wild card entry. Why they are telling that epigenetics is a wild card entry? Because it is heritable changes that do not impact on the gene sequence. So if there is a slight modification in the gene site or histone proteins like methylation, phosphorylation or acetylation or ubiquitination, this may modify and production of microRNAs. So this microRNAs is the target of our proteins. Okay, this microRNAs is the 22 base pair non-coding RNA sequences which is roam around in our cells. Okay, cellular metabolisms. So what is this microRNAs functions? This microRNAs binds to the normal mRNA. So once it binds to the mRNA and normal protein synthesis may be inhibited, the regulatory proteins or another proteins, whatever the protein which is necessary for our cell signals, that will be blocked. So that is the major function of this microRNAs, leads to a disease. So that's why they say as the epigenetic is a wild card entry, because the modification in histone molecules leads to the production of microRNAs. How we can control the microRNAs? By intaking of a good nutrition. The nutrition play an excellent role in epigenetics, because when you take in good fresh vegetables and fruits, a good nutrition, this can modulate the DNA methylations, and this DNA methylations inhibits the suppression of the microRNAs. So if there is no microRNAs, the translation of good proteins is a good thing. Okay, so where is the problem here? If there is insufficient nutrition and enzyme is insufficient, that leads to poor function of our genomes. Okay, so if there is any insufficient nutrition and enzymes, leads to a poor functions. So what we have to do? How we have to tackle this one? Our Indian culture, we have a traditional spices. Nowadays we are converting to the Western culture, but our traditional spices is a cancer signaling modulator. So daily consuming the our traditional spices. For example, cinnamon, cardamom, coriander, curcumin, cumin. These are the most important spices which is having an excellent bioactive component and which is helpful for our cell signals and protecting our genomic sequence by the mutagens. Okay, so for example, these phytochemicals, this traditional food have a phytochemical like resveratrol, curcumin, flavonoids, polyphenols like epigalactosin, okay, caffeinic acid, many things. So the mutations have a good interactions with the genomes. So for example, omega fatty acids and fatty acid from fish plus folic acid, vitamin B12 and B6, okay, and minerals like magnesiums, zinc and selenium, and the plant components like genistein, curcumin, resveratrol, flavonoids, plant polyphenols, epigalactosin. These are the good nutritions which have a good interactions with the genomic sequence and protect us from the dietary mutagens. Okay, so I cannot explain all this thing. So let's we see about only a few like only one plant bioactive components which is working in our laboratory. So we have worked on flavonoids and cancer biology. Okay, so the flavonoids, how the flavonoids remodeling and cancer acting as a cancer preventing agents. Okay, the genetic alterations always triggers the cell signaling pathway by transforming a cell cycle of normal cells into a cancerous cells. So the flavonoids are abundantly presented in natural foods, which can protect against the age-related disease by protecting the DNA, lipids, protein molecules from mutations and oxidative damages. Okay, so it can target as a multiple targets. So what we can do is the flavonoids targets cancer cells by altering the gene expression, maybe by direct interaction with the radical suppressions of the carcinogens, which is produced by. And mostly the flavonoids act as a free radical scavenger and hormone regulator, protein regulator, targeted drug and modulating the cell signaling cascade. Okay, so how it is modulating the cell signaling cascade? The cancer means it is an uncontrolled cell proliferation, continuous proliferation of the cells plus inhibition of the cell death. That is the major signalings for the cancer cells. So these flavonoids inhibiting the cancer cells by inhibiting the activation of epidermal growth factor receptor pathway, JAK-STAT pathway. There are so many pathways for cell proliferation. Okay, so all the pathway have particular some cytokines and cell signaling enzymes and cell signaling genes. These flavonoids block the cell signaling genes, the MAP kinase pathway, PI3K kinase pathway, all the pathways. Okay, it was reported. So by blocking this pathway and regulating the cell signals, okay, and inducing the cell death, that is also the major impact. For example, the flavonoids, if the flavonoids block the topoisomerase and the DNA replications has been inhibited and simultaneously it inhibits the cell proliferation. If the flavonoids blocks the RAS and MAC pathway, like this NF-kappa B, okay, so it activates the NF-kappa B is the activation of BCL2. What is the function of BCL2? This BCL2 is inhibition of apoptosis. So always, if once the cell is produced and the cell have to die after some time. Okay, but here if this gene is BCL2 is activated, that inhibition of apoptosis, the cell cannot die it own. So what I have to do is we have to block the BCL2. So that is the major impact of the flavonoid. The flavonoid blocks the BCL2 and further gene proliferation has been inhibited. Okay, so how it is inhibited? the flavonoid induce the cell death receptor, okay, by encystic pathway and excistic pathway. The flavonoid activates the caspase 8, caspase 9 and caspase 3 pathway and fragments the DNA into a small components, induce apoptosis. And another way, the flavonoids also simultaneously induce P53 and activating the caspases 9 and caspase 3 pathway to induce the apoptosis. And another encystic pathway is it blocks the BCL2. Okay, once it blocks the BCL2, automatically the BAX gene is activated. Okay, once the BAX gene is activated, there is an intracellular ROS and mitochondrial membrane potential and intracellular calcium is increased in the mitochondria that leads to release of cytochrome C from mitochondria. So once the cytochrome C is released from the mitochondria, that leads to activation of caspase 9, leads to caspase 3 activation and induce the apoptosis. This is the major function of the flavonoids. So we have to take our traditional food items which have high content of flavonoids and polyphenols which can helpful for our cell cycle and modulate our cellulatory mechanisms. Okay, so the another one is bitter gourd, but this bitter gourd is the one which the people hate in their life, but it is a very good protector of the modulator hepatoprotective mechanisms. It have a very good metabolic regulation and inflammation radioprotective mechanisms. It have a more good components. We are also working on bitter gourd in our research laboratory. It is also have a metabolic abnormalities, therapeutic efficacy to combat against the malignancies. Okay, importantly, this have a bioactive component which can target the metabolic disorder and molecular target of specific site in signaling cell signaling cascade, and it can pave the way for fruitful metabolic disorder and tumor preventions. Okay, for example, a hyperactivity of histone deacetylation can be blocked by bitter gourd because bitter gourd have a MCP30 molecules. This molecule blocks the histone deacetylations. Once the histone deacetylation is blocked, it can activate the apoptosis process. Okay, so the loss of VGF angiogenesis, loss of adhesions, many things, the functions. So the cancer initiation has been inhibited. So this is the more good and excellent activity of the bitter gourd. Okay, so the another thing is it have a wild functions. Okay, so various functions it can have an against the cell cycle regulatory mechanisms, as well as induce the caspase 3 and blocks the BCL2 and induce the gene of caspase 9, as well as MAP kinase pathway also simultaneously. And it have two type of thing. It induce the cell proliferation as well as apoptosis. It induce cell proliferation in beta cells do in the diabetes conditions. If the people have diabetes, if they are taking this bitter gourd, it modulates the MAP kinase pathway and NF-kappa B signaling cascade and increase the lifetime of pancreatic beta cells. Okay, so it inhibits the apoptosis in pancreatic beta cells. But in cancer conditions, it alter the signals in opposite way and induce the apoptosis pathway. Okay, so by induction of cell cycle or caspase, anything maybe. Okay, so that's why the bitter gourd is the modulator for the cell signalings. Okay, so overall, when you have the phytochemicals or phytocomponents like honey, green tea, soya beans, these are all alter the inflammatory reactions by suppressing the oxidative stress and gene alterations. That genetic alteration leads to result in a proteomic alterations. So once the genetic alteration is good and the epigenetic damage and DNA damage may be altered, the inflammatory reactions and gene alterations together it can that can designs to the experience can associated with diseases. However, only a few rare diseases like as Huntington, Toy-Sage disease, or a certain sleep determine the genetic makeup. Okay, the most gene have a flexible expressions, and researchers found there is a complex interactions among the multiple genes according to the environment and diet. So the single nucleotide polymorphism may be based on a nutrition status or interacting with the single nucleotide polymorphism or stress or environmental or lifestyle choices. Okay, so we have to find which is the one and how we can protect ourselves. Some people they may have certain SNP and higher level. Some people may be certain nucleotide polymorphism at lower level. It may be including in the ethnicity or gender variability. So this may be resulted in the clinical representation of familial risk or prevalence of significant some allele variations. This may be population of a specific. The patients may have additional risk. It is not to measure by the test. So negative finding also not imply that patient can take a risk. There are certain genes. Okay, so first we are going to check about the methylene tetrahydrofluoride reductase. This is the enzyme gene which is responsible in the methyl transferase synthesis of folate molecules. Okay, you know about the folate, right? The folate is the major molecules involved in the DNA synthesis. Okay, and this is the cycle that how the methyl function is involved in the methylene tetrahydrofluoride reductase is involved in the function of the folate cycle. Okay, so here we need a specific vitamins and folate molecules. So if there is any nutritions not able to give a properly, then there is a defect. This is we can tell is as one carbon atom defects. Okay, because here one carbon atom unit is able to have the transfer depends upon the nutritions. So if there is a folate, vitamin B6, vitamin B12, riboflavin, vitamin bennettin, these are the important nutritions used for synthesis of homocysteine. Okay, this homocysteine, if it is high or if it is low, it is the problem in the methyl transferring. Okay, and affects the folate synthesis. So this folate is the more important thing in the DNA synthesis. Okay, so this is the major thing. So daily in our consumes we have to take this food items will be a good. And another thing here, how it is involved in the SNP, single nucleotide polymorphism, because there is 2 copies of methylene tetrahydrofluoride reductase gene. Among this, the C677T is the major impact. Okay, so first we have 3 type of genomic sequence. One is some people they have wild type of genomic sequence. When they have the wild type, that is negative wild type, they have the full strength of the enzymes. That type of the people, they don't have any issues. But if they have a heterozygous, okay, some their enzymes are not completely active. Some enzymes, like a few 50% of enzymes may be active, but 50% of enzymes not be active. Okay, if there is homozygous, okay, then the enzyme is reduced. So 60 to 70% of the people, 70% of the enzymes not active. So in that case, mostly the C677T, this is the disease risk of the most pronounced homozygous. So we have to analyze the genotype, whether they have the wild type or the heterozygous or the homozygous. According to the single nucleotide polymorphisms, they can able to find out or figure it out which type of nutrition is good for the certain people. Okay, so according to that, the nutritions and genomics it will be work on. So here the homozygous and heterozygous, it is resulting in reduced methylation, so that leads to risk of the enzymes. Okay, so if there is a reduced methylation activity resulting in Alzheimer's disease, anxiety, cancer, cognitive decline, depression, and heart disease or stroke, neuronal tube defects, it's maybe any conditions. These are the conditions if there is a defect in the methylene tetrahydrofluoride reductase defects. Okay, so for this, how they can given? So the people can, we can have to detect whether the people have the biomarkers level is high or low. That biomarker is homocysteine. The homocysteine is the major biomarkers. Okay, so once the homocysteine having issues, then we can able to find out what is the defects. Okay, so if there is a homocysteine level is high or low, there is an increased risk of homocysteine, especially when there is lower level of B vitamin, mainly folate. That leads to the disease. Okay, several studies also suggest that lower folate level and moderate total homocysteine elevation may be correlate with hypomethylation in DNA of lymphocyte. Okay, so this leads to vascular problem that can associated with hyper homocystinemia that may be partial due to DNA hypomethylation. Okay, so what is the lifestyle? What is the therapies we can use for this if someone have this type of single nucleotide polymorphism? So we can ensure that adequate intake of dark green vegetables or other vitamin B vitamin rich food which can protect themselves and given a high methylation activity. So it will be protect for the people who have the C677T genome sequence. Okay, so we can given the extra supplementation along with folic acid or B2, vitamin B2, B6, B12 and betaine, anything. Okay, but they have to change their lifestyle, because some people, the people who have this genome sequence C677T, and if they have continuously drinking, heavy drinker as well as smoking, and this will inhibit the methionine synthesis process in the DNA. Folate, it leads to folate depletions in mitochondria and abnormal DNA synthesis that resulted in DNA methylations. But they have to change their lifestyle. Once they change the lifestyle, it will be good for these people. So they have to analyze the single nucleotide polymorphisms to along with the nutritional diet. Okay, so this is the snapshot of overall view about the methylene tetrahydrofluoride reductase enzymes activity. Next one is the catechol O methyltransferases. This one we can say as a neurotransmitter. It is an important thing, and this catechol O methyltransferase enzymes, this ensure and catalyze the movement of methyl group from S-acetyl methionine to catechol or catecholamine. Okay, so this catecholamine is there. It induces dopamine, epinephrine, norepinephrine, estrogen, and various chemicals and toxins. So what we have to do is, it is the major thing used for better health for life, that is BH4 cycle also, or it is a tough cycle also. Okay, so this catechol O methyltransferase gene has well studied and have a common variant at the codon of 158, but the allele is different. In this 158, valine is there. It have a greater activity of comet. Okay, but if there is a methionine allele, substitution of valine, then it is a problems. Okay, so that reflects if there is a homozygous, then it affects an increased risk of ancient women for the breast cancer and but marginally there is a decrease in the conscious ones. Okay, it reduce the pain thrush and exacerbate the one experience of the pain, as well as increase the risk of the chronic pain syndrome. And mostly in the male, it increase the fracture risk. Okay, but greater the bone mineral density response and physical activity we can in helpful for the people. If they do the more physical activity, it will increase the bone mineral density. Okay, and if there is a homozygous thing, and there is a possible risk of the people can addict to alcoholism. Okay, as well as the possible risk of Parkinson disease. This is a reported one. If the people having the homozygous gene, there is a possible risk of alcoholisms and a possible risk addict to the alcoholisms and Parkinson diseases. So in this case, what is the treatment option? So minimize the stress to keep the catecholamine level low. If the people is having methionine, okay, if there is a valine, there is no risk. If the people is having methionine in their single nucleotide polymorphism, then have to minimize the stress level. Okay, and ensure adequate intake of vitamin B6, B12, folate, magnesiums, and methionine to support the adenosyl methionine and prevent the elevated level of homocysteine. Okay, that can inhibit the adenosyl homocysteine activity. Okay, that comet activity. Okay, this preliminary finding can also reduce the cardiovascular disease by intake of the vitamin E. And once if they intake the vitamins and minerals, that can intake this which have this type of genotype, it can suppress the selective serotonin uptake and inhibit the antidepressant. This is the major thing we can able to find it. So this is the major treatment options. Okay, this is the major enzymes involved in all the inflammatory things. So we can say as inflammatory markers or inflammation monster. Okay, so this is TNF alpha, tumor necrosis factor alpha. This is a balance key. Okay, so if the tumor necrosis factor is upregulated, it is good. Okay, the tumor signal is good. It is good for the cancerous cells to die. If it is down-regulated or if it is higher level of the tumor necrosis factor, okay, so that can be inflammatory response for healthy cells. It gives the inflammatory response to the healthy cells and converted to the bad cells. Okay, so the tumor necrosis factor is the pro-inflammatory cytokines secreted by activated macrophage, and it have an important role in defense against the infections. And however, release of cytokine increase the oxidative stress and inflammation, mostly obesities, metabolic disorder. Okay, so it has promotes the identified and may regulates the TNF expression. In sometimes, if there is in the position in the polymorphism, TNF alpha polymorphism 308, instead of glycine, alanine is there, then it can affect the cytokine productions and associated with regulation of TNF expression. Okay, so it interfere with the transcription factor binding site and other regulatory elements. So it gradually increases, that leads to risk of inflammation and oxidative stress. At the same time, the clinical issues also we can see in the heterozygous genotype also. And if there is an increase of TNF alpha, risk in increase of osteoarthritis, as well as elevation of acne vulgaris among the conscious ones. Okay, so what is the options? It is an abnormal fat loss, visceral fat produced TNF alpha and interleukin 6, and weight loss is mostly associated with decrease of these inflammatory cytokines. They have to do more physical activity with less calories food, which can helpful for their nutritions, which can helpful for the expression of TNF alpha, and they can improve the insulin sensitivity level and control the stress response. Okay, so this is the major thing we have to do. And they have to the nucleotide polymorphisms, which they have higher lower level expressions, the weight gain, abnormal cholesterol profile, and can high intake of saturated fat and fatty acids. Okay, so fish, vitamin E, fish oil, and N-acetylcysteine, green teas, and lactobacillus, estrogens, if they intake, it is the possible thing to control the tumor necrosis factor. Okay, this is the snapshot or summary of this tumor necrosis factor alpha. So overall, when we come to know that the single nucleotide polymorphisms and can involve with the nutritions, can compromise the epigenetic level of our genome, and can apolipoprotein, TNF alpha, that catechol O methyltransferases, methylene tetrahydrofluoride reductase. These are all is involved in the disease, metabolic disorder, neurodegenerative disorder, which can suppress by uptake of vitamin E, omega-3 fatty acids, vitamin C, and vitamin E. Okay, so this is the major thing we can have. So overall summary, there is a lot we know about our genes, but there is a lot still we don't know what is the function of gene, how it is modulating in a epigenome, and what are the food is modulating our genomic functions. Our lifestyles changes in the genomes. There is still we have to know about the nutrigenomic applications to clinical medicines, but there is some recent data is emerging. They are giving us suggestions that study for the single nucleotide polymorphisms, according to that giving the nutritions balanced. Okay, if sometimes if the nutrition is high also it is a risk, and if the nutrition is low also it is a risk. Eating a food, good food, and it is a good signals. Okay, the food is the contains a full information signals to deliver to our cells, because the signals transfer to the genome, and genome giving a good signals to the cells. So the nutrigenomic testing should be coupled with the laboratories and nutritional assessment for the clinical applications, and we can go for that the complete picture and assessment. We can find the important genes and their alleles, which is presented according with that clinical therapeutic strategies we can improve. So we can make a new laboratories and new technical diagnostic laboratories with new skills, finding the single nucleotide polymorphisms according to given a suggestion of nutritions, which can helpful in the clinical therapeutic conditions. Okay, so this is the epigenetic we can modulate with our nutritional conditions, nutrition intake. Okay, so if anyone have doubt, you can ask me regarding this topic. I can ready to explain about this. Thank you for this presentation, listening my presentation. Hello.
Yes, ma'am.
If anyone have any doubt, you can ask me. Okay, thank you. And maybe time is very less, I think so. So we can go for the another topic, okay? If you have doubt, you can ask me or message me. If you need my presentation or if you need any questions, you can call me. No problem. I can give in my number and email, and we can have some doubts. I can able to clear you regarding the work.
Thank you very much, ma'am, for sharing your valuable knowledge on epigenetics. We are proud to have such a wonderful alumni, ma'am, also.
Thank you. Thank you.
Now let us move on to the next session. The aim of education is the knowledge, not of fact, but of values. It is an occasion like this we get opportunities to test our knowledge and understanding. Now let us move forward for the next session. In order to introduce the chief guest, here comes Mr. P. Palberman, Assistant Professor of our department. I welcome you, sir.
Very good afternoon to everyone. I greatly honored to introduce our resource person, Dr. S. Vignesh, Associate Professor, Department of Microbiology, Indian Institute of Food Processing Technology, Tanjavur, Tamil Nadu. He received his master and doctoral degrees in Bharathidasan University, Tiruchi, Tamil Nadu. He has published numerous research articles in peer-reviewed journals and book chapters. He has guided more than 20 PG and 10 MPhil students. He has also presented several papers in national and international conferences. He serves as a reviewer in many international journals like Elsevier, Frontier in Environmental Science, and etc. He has organized a national level workshop and conferences. He specialized in food and environmental microbiology, quality control aspects, nanotechnology, biokerosene studies, bioremediation, heavy metal studies, and etc. I feel very happy to welcome and introduce such an eminent person to this international virtual seminar on nutrigenomics. And also, I thank you for giving a nice opportunity to this virtual seminar. Once again, I welcome you, sir. Thank you.
Thank you, sir. Am I audible? Can everyone hear me?
Yes, sir, audible.
Yeah, fine, sir. You can see this display, my presentation.
Yes, sir. Please put full screen, sir.
Yeah, is it okay now? Is it okay now, sir?
No, sir, not displayed in full screen, sir.
Yeah, I put it in the full screen actually. Is it okay now?
No, sir. Is it possible to send this PPT to our mail, sir, so that we can present it?
Yeah, okay, fine.
The mail ID is microbiology@vhnsnc.edu.in, sir.
Just like that.
I'll be typing it in the chat box for your
No
reference.
problem. You already send me the mail actually.
Yes. Already. We have sent the mail from that
Okay, no problem.
mail ID itself.
I will share.
Okay, sir. Thank you.
Yeah, please check your email.
So please let me take a few minutes, sir. So we humbly request you to wait for a few minutes, sir.
No. Shall I share my PPT?
So we are ready, sir.
Yeah,
Shall we
fine.
start?
Yeah. So, respected principal, college management authorities, and dignitaries, beloved friend Dr. Balasubramaniam, my dear colleagues, dear students and participants, a very good afternoon to all. Today, I'm like a night watchman. When you are playing a test cricket, the last session of the day, if any batsman will get out, the captain and the coach will send one bowler to Greece. That means they have to manage something. So I think today I am the last batsman of this program. Anyway, I only prefer this slot due to some of my other works. So Dr. Bala arranged very well. So first of all, I would like to thank Dr. Bala and other team members who have given me an opportunity to share my thoughts and views with the microbiologist. So go to next slides. So today we are going to discuss about role of gut microbiome in nutritional uptake. As you know very well, the microbiologists are like a monk. Cricketer Murali Vijay pet name is Monk actually. You know why both are same? Why I'm telling like both are same? You know. So both are always dealing the invisible matter and the inevitable matters. And their main role is they have to bring the invisible matter into the limelight. So that's what microbiologists are always doing, because we don't know what are all the microorganisms which is present on our body or inside our body or in the atmosphere. But our duty is we have to identify, isolate, and identify that organism that is an invisible matter. That is, we don't know what kind of microorganisms are there. But within the matter of few hours, nowadays in the latest technology, within the matter of few hours, we can easily identify the organisms. So that's what I'm telling. The microbiologists are like a monk. So next, go to next slides. So in the basic information today, as you know very well, our fecal matter contain lot of microorganisms. Normally 5 to 25% of our fecal matter composed by the enteric organism. The enteric organism is nothing but gut microbiota. The gut microbiota, so normally in microbiome, what we can define the microbiome, that is exactly, we can say like it is not only mean the microorganism, it is apart from the microorganism, entire habitat, in the genes and the surrounding environmental condition, everything is, it is encompassed the habitat, microorganism, genes, and the surrounding condition. That is we call this microbiome. This average, the individual human being weight is like a 70 kg. That person may contain 100 trillions of microorganism, and its weight is like a 200 gram. It is like a small apple or small mango, so like something like that. And you know very well, as per Broadstall stool experiment, they mentioned very clearly 1 gram of our fecal matter contains 10 power 9 to 10 power 12 microorganism, including bacteria, virus, fungi, major fungi or minor fungi like mold or yeast or whatever. Approximately it contains 10 power 9 to 10 power 12 microorganism. If the normal if this is for normal human being, if a person is affected by the any infection, bacterial infection or fungal infection or any other thing, especially bacterial infection, it may go up to 10 power 14 or 15. So that's what in FSSI they are preferring the VIDAS tempo reader and the tempo filler experiments. In that it is working under the principle of MPN, and we can identify the organism up to the 10 power 15 level. For the manual, we can identify up to the 10 power 9 actually. Apart from beyond that limit, beyond that level we can't go, but we can use the tempo reader and tempo filler for the identification. And as you know very well, introns and extron sequences is still there in the bacteria as well as in the human being. And nearly 150 to 200 times more genes in the individual microbiota than in all of their other cell put together. And when compared to the human beings, the microorganisms are over 3 million genes are there, which is present in our body. That's what I represent. The human cells are like 30 trillion, but the microorganism which is present inside our body, that is more than 1,000 trillion. That in terms of genes, the humans are more than 99% microbials. In gut microbiota, it is a richness and diversity and stability over the time are key features of the health associated human gut microbiome. It is like your fingerprint. Gut microbiota composition is unique to each individual. That is very, very important. And the gut microbiota can be easily affected by the external force like a pH or a temperature or the other conditions. Like, and while some are the less controllable like genetics, delivery method, geography, aging, and you can act on your diet and lifestyle to help your gut microbiota work properly. And your gut is more than a tube, it home to more than 1 lakh trillion microorganisms that participate in the digestion part that we are going to discuss today. And the different functions of the gastrointestinal tract, as you know very well, when we are taking normally in our home, our mom or our ancestors, our grandpa or whoever, they are preferring like if you have suffered by diarrhea or whatever, and they are telling that, go to next slide, and they are simply telling that you have to take a curd for any, if you are suffered by any diarrhea or dysentery or whatever, they are simply preferring and they are suggesting you to take a curd. The curd contains a lot of LAB organisms. So what is the difference between LAB and the harmful microorganism? Definitely it will reach to the intestine. Whatever organism we are taking orally, whatever food or water we are taking orally, finally it will reach to our intestine. In our intestine, this kind of beneficial organism will easily merge with the mucous membrane, and first it will reach into the mucus, and it will not affected by the mucus. Then it will reach into the mucous membrane, and it will adhere on the mucus, that is epithelial cell. But in the case of pathogen, you just imagine if the pathogen is entering into your body, if it first it will reach to your mucus, then it will be escape from your mucus. That is, a mucus contain lot of enzyme. Apart, then it will reach into the mucous membrane, then it will cause the inflammation, then it will enter into your cell. So then only your macrophage will activated and phagocytosis process will be done. If the phagocytosis process is failed, that means the organism is very virulent, then only it will involve in the proliferation and the multiplication process. Then it will cause some infection, and it will produce some toxins or secreting the EPS, that is a LPS or EPS, exopolysaccharide or lipopolysaccharide, or some glycoproteins. It will release some primary or secondary metabolites that may cause infection, or the organism itself contains some of the like a cell wall contain S layer or slime layer capsule. It contains the antigenic nature, and it can cause the infection. Okay, so what do you eat does not only nourish you, it also feeds the trillions of microorganisms that live in your gut, including prebiotic fibers, that is a fermentable polysaccharide, dietary fiber. That's we call it as a dietary fiber is rich in the like spinach, okay, kirai எல்லாத்துலயுமே நிறைய dietary fibers இருக்கு. That is a prebiotic fiber and fermented foods with probiotics as you make progress on the path towards a healthy gut. How this gut microbe evolve? So that is a very important one. So when born vaginally or cesarean, okay, at any way, if the baby born vaginally, an infant is colonized by the mother's gut and vaginal microbiota first. And if it is a cesarean case, then that is directly exposed to the skin and hospital environment. And the skin means first the baby is touching. The baby is the mother is always carrying the baby, so it can be easily transferred from mother to baby first. And hospital environment, don't confuse yourself, that is a nosocomial infection is different, that is a hospital acquired infection. When we are talking about infection, that is different. Next slide. When we are talking about nosocomial infection, that is different. Here we are talking about hospital environment microbiota first. Okay, the hospital environment, the microorganisms are present in our surroundings, so it can be easily transferred into the body. And the mode of feeding, breast or formula milk. Formula milk is nothing but it is a pro none or none which is available in the market, and it is a not exactly, it's the milk is not come from the human being or animal, it is a formulated one. Okay, so it has a major impact on the composition of infants microbiota. Like breast milk is not only contain the live bacteria, especially bifidobacteria, but also a great variety of complex carbohydrate like human milk oligosaccharide, HMO. We simply call it as HMO. Next slide. HMO that cannot be digested by babies. In turn, other in complex carbohydrate prebiotics, by influencing the composition of bacteria in the infant gut, especially two organisms are very, very important. That is a LAB organism, and another one is bifidobacterium. Bifidobacterium is very, very, very unique, and this bifidobacterium is always involving in the digestion of HMO, that is a human milk oligosaccharide. Okay, so it is similar. When the infant will take formula milk, that microbiota is almost similar to when the infant is taking a breast milk or cow milk or whatever. And it is also resembling to adult with the higher overall bacterial diversity. But bacterial diversity will be vary, as well as depending on the latitude and the longitudinal variation, spatial distribution, the microbiota diversity as well as the load will be vary. But when we are talking about the disease causing nature, when we are always say that three important factors are very, very important. One is the pathogen must be a virulent, and second one is load. How much load you are consuming, how much load it will enter into your body, and third one is a host immune system. Based on these three factors only, you may be suffered by some infection like that. Here we are telling there's a likewise here the beneficial load of beneficial organism, load of that commensal organism will always be vary from the different areas. And how the microbe will transfer from womb to baby, that is a mother to baby. which is started, this process is started the baby which is present in the womb, mother womb. That is the microbial colonization of fetus may drive immune programming prior to delivery. That is already started. Still people are working on it. Through the placenta only, it can be transferred from gut region to mother gut region to baby, and bacterial translocation from gut to placenta. So this is also one important one. And finally, the bacteria and the bacterial metabolites will be translocated to breast milk from the maternal gut. So this is one of the pathway. Apart from that, the milk contain lot of things, immunoglobulin. Okay, so that is a different one. And the main function of the bacterial metabolites up at the maternal fetal interface prevent epigenetic changes impacting fetal immunity. That is, fetal immunity is nothing but SCFA production. SCFA means short chain fatty acid production from the maternal gut. So that's what we are telling. Already I told you, while it is a cesarean case or whatever, apart from the vaginal, apart from this, through the skin, it can be easily enter into baby. And the candida will also enter into baby very fast, and it will grow rapidly. So within the matter of 3 to 6 months, it is equal to firmicutes and bacterioids. The fungal, the candida, especially the candida, will be dominant in the baby actually. And through mother, oral, placenta, gut, and vaginal. Based on these 4 routes, the microbiota will enter into the baby. Next slide. So effects of the maternal microbiota in the pregnancy and the early life. So as you know very well, just now I told like breast milk is a very important one, and it can be easily, the gut microbiota will transfer from mother to baby, maternal microbes, nutrients, antimicrobial peptides, and this can be easily, next slide, this can be easily transferred from mother to baby, and that is the starting point. And apart from that, when vertical transmission of maternal, vaginal, and fecal microbes, here you can see the bottom of that figure, vertical transmission of maternal and vaginal and fecal microbes. Okay, so this is a very, very common point. Mostly the candida and enterobacteriaceae family will easily enter into the baby through this route. And fetal immune and gut mucosal development, it is happening in the early stage, and transplantal signal from the maternal microbiota, and the seeding early life of microbiota, that is a common one up to the 3 months. It is very vigorous, and early life immune development and neurodevelopment, susceptibility to childhood illness, including infection, asthma, obesity. It is a very, very common one. If the microorganism is not properly harboring, it is not properly regulating your gene, so definitely it will cause some infection like obesity. That is a dysbiosis. We call it as dysbiosis, obesity, or immunological problem, or some other problems. And both bacteria and fungi play a very important role. Apart from the Enterobacteriaceae family, Bifidobacterium, and bacteroids, Lactobacillus, and some other organism, especially in fungi, Candida play a very, very important role. And next to that, Rhodotorula, Candida is a very important one, which is a vaginal microflora, and normally which is present in your buccal cavity, especially the white color patches. When you are wake up in the morning, you can just feel that when you are rotate your tongue after you wake up in the morning, the patches will be accumulate on your teeth. That is a white color patches. You can just take it out. That was identified. That is the second sample which was collected by the Anton von Leeuwenhoek. First, he have collected. First, he he coined the term animalculus. The first time, he have collected his own fecal. Second, he have collected a sample from his own buccal cavity. So that is the thing. The Streptococcus mutans and the Candida play a very important role. In market now, people are telling like you can use our toothpaste and whatever. And if you are using any toothpaste, nothing will work out more than 3 hours, because definitely we are taking some water, we are taking some food. So microorganisms are easily enter into our body through this oral way, so that it will easily harboring in your buccal cavity first, especially this Candida, and it will create that white color patches. So this is the way the biota will be easily entered into body, and it will be stored in your gut region. And predominant, that is a firmicutes, that is a lilabi organism, and bacteroids, that is nothing but bacteria. That is a bifidobacterium. That is a bifidobacterium one. And Verico microbiota. Next slide. Verico microbiota is a very important organism in that phyla. Very important organism is Akkermansia. Akkermansia is a mucin degrading bacteria. Very, very important gut microorganisms. And these are all the organisms play a very important role in the gut region. Even though this general profile remain constant, gut microbiota exhibit both temporal and spatial differences. That's what we discussed. And especially, and you can see that when you are taking the microorganism through your esophagus region, that may be a 10 per gram of content in the esophagus, and it will end the stomach. Then it will reach to 10 power 12 per gram of content in the colon and distal gut. Distal gut means in the gut region, especially. So in the gut region, the processing will be start in the stomach, and it will reach into the smaller intestine, and then it will reach into the larger intestine, that is the colon region. Then it will go to rectum, then it will release as a fecal matter. The lactic acid will be added in your nutrient, then the color will change as a yellow color in your fecal matter. Here, during this process, lot of organisms are mixed with your nutrients, and automatically once the nutrient will be there, the proliferation is very fast. Mostly the gut microorganisms are phosphidious microorganisms, especially LAB and Enterobacteriaceae family, and other organisms like bacteroids and other organisms. The generation time of these organisms are less than 1 hour. So the growth is very rapid, and it will grow very rapidly, and it will be accumulated in the fecal matter, and it will also first accumulate in the inside of the intestine, and it will allow the organism, it will help to the nutritional uptake, that we will discuss later. So as we said, like predominant microorganisms like bacteroids, bifidobacterium, streptococcus, enterobacteriaceae, and enterococcus, clostridium, lactobacillus, ruminococcus. So these are all the organisms are very, very important, and these are all the organisms are involved in the fermentation process, that is a polysaccharide fermentation process, and involving in the digestion process also, so that is resulted in the synthesis of SCFA, that is a short chain fatty acid. Then it will be converted into butyric propionate and acetate. Then it will involve in the glycolysis pathway. Next one. Next is it. And some of the factors are shaping the gut microbiota. So as you know very well, the mode of delivery, either vaginal or cesarean, and diet during the infancy and adulthood, like breast milk or formulated feed, and adulthood, vegan based or meat based food. So based on the food, it will be definitely the microbial load and diversity will be vary. Next slide. And the final important one, apart from the pH, temperature, that is different. Here we are telling some other important factors, and very important one is antibiotics. As you know very well, antibiotic like molecules that are derived from the environment or in the gut commensals community, that only involved in the decoy process, that is a mimic process. Like mimic means the receptor will be there, instead of pathogen, this prebiotic will be attached on the receptor, and that is called as a decoy. So it will be act like a mimic, so that the pathogen will not bind on the receptor. If pathogen will bind on the receptor of that mucous membrane, that is the epithelial of the mucus, epithelial cells of the intestine, definitely it will cause an infection. It will cause an inflammation, and it will cause an infection. So that it will prevent it because of this prebiotics. Prebiotics is nothing but whatever food you are taking, especially the very good example is HMO. Here, through the slides, I am just giving some examples of them. HMO. Okay, so that will be easily understandable. That's why. And here we are talking about the third important point is antibiotics. So antibiotics, you know very well, nowadays we are using different antibiotics, different generation of antibiotics, and that antibiotics are almost cephalosporin and chloramphenicol play a very important role. And most of our cultures, most of our strains which is present in the normal environment, mostly our strains are resistant to at least one antibiotic nowadays. Because of this horizontal transfer. When we are talking about the vertical transfer of the microorganism from like mother to fetus, in the previous slides we have discussed. Here we are mentioning the horizontal transfer of resistant genes. As you know very well, within the matter of 20 to 40 minutes or 1 hour, within the generation time, automatically the gene will be transferred from one organism to another organism. So that is especially resistant gene or gene, resistant gene. So it is a conjugation, through conjugation process. When we are talking about this kind of resistant genes, that is asexual. That one is a transformation, because we have to make our cells as a competence. Competence means it is the cell is ready to uptake the genes from the outside. That is a transformation process. Here, transformation process is almost, it is very difficult. Here, the horizontal transfer is a very, very common one in the gut region, because a lot of microorganisms are there, and it will be easily involved in the conjugation process, so that it can be easily transferred. So that what will happen when we are frequently using the antibiotic without doctor prescription in every home, we know very well, in our packet, in our wallet, we are always keeping the medicines. In our home, our mother or our daughter, our sister, fever or cough or headache or whatever, they are readily having some pills, they will immediately give it to you. What will happen? Finally, if you are not properly taking the course, like 3 days or 2 days, definitely the organism, if you are not properly taking the pills, the organism is resistant to that particular antimicrobial. The antibiotic will enter into your body, and it will create a selective pressure on your organism, so that the organism is slightly change itself, and it can be able to survive in the extreme environment. And finally, it will be released through your urine, fecal matter, and it will also reach into the aquatic environment. Finally, the microorganism will be evolved. The antibiotic resistant organism will be evolved. So that what will happen, based on when we are using the antibiotic, automatically the microbial load will be vary, especially the gut microbiota will be vary. Next slide. So gut microorganism, as you know very well, Lactobacillus, Streptococcus, Helicobacter pylori. So these are all the organisms are very, very important. Apart from that, bacteroids, Clostridium and Bifidobacterium, and Enterobacteriaceae family, especially in the colon region. The organism level is more than 10 power 11 to 12, and the pH is also very favorable to the microbial growth, that is around 5 to 7. Next slide. So it will be there. These are all the organisms are involving in the nutritional uptaking process. Like especially when we are talking about the ascending colon, which is present in the right side of the colon, that is the large intestine is called as ascending colon, and the small intestine one is left side, the not small, that is a left side colon is called as a descending colon. So both are having some bacteroids, lactobacillus, LAB organism, and bifidobacterium. And apart from that, this colon contains D-sulfomonas and D-sulfobacterium. This is a very important organism. These are D-sulfobacterium, D-sulfomonas is a very important organism which involved in the sulfur cycle, especially the Bacteriaceae family and some other Gamma Proteobacterium and Clostridium organisms will be always present in the small intestine. That's what we observed so far. Okay, so which is present in the small intestine. When the nutrient our food will come from the small intestine, and it will come to the large intestine. Along with that, the nutrient, not only the nutrient will come from small intestine, lot of microorganism will also come to the large intestines. So this is the way it will be transferred. And the next one. So lot of gut microbiota and beneficial and harmful microorganisms are there, and the activities will be different, like bifidobacterium. Just now I have listed out. Next slide. So some other organisms are also considered as a evil. When you are taking a yogurt or probiotic, definitely it will good to your health. When you are taking some other food, fried or some other meat or whatever, sometimes what will happen there. So mostly the foreigners are taking the raw meat, and raw meat in the sense of raw oyster and some other marine foods. They are taking the raw meat. So what will happen? That contain lot of microorganisms. Especially in the epidemiological, we have a strong epidemiological report about the raw oyster eating. That is the salmonella is the prime organism. Meat and egg, the salmonella is the prime organism. and more than 30% of the people is affected in the foreign region while they are taking the raw meat, affected by especially salmonella and Escherichia coli. So this is a common one. So these are all the organisms are easily present in the some other foods. When we are taking the yogurt or probiotic or some other beneficial, that is some other foods, some foods are having contain a lot of LAB organism that is good to your intestine. And the role of gut microorganism. Now we discuss about the role. Here, the gut microorganism, go to the next slide. So defense, first one is it is involving in the defense, and it is a tolerable one. Whatever food we are taking, it can be easily tolerable. It is acidic nature or alkaline in nature, and it is a very difficult or whatever, it can be tolerated, and it is directly involving in the digestion and the absorption process. Next slide. Absorption process, and very important thing, it is entered into the metabolism. It is directly, next slide, it is involving in the metabolism. Metabolism means when the fermentable polysaccharide will enter into the mucus, that is the inside of the intestine. Next slide. Enter into the intestine, definitely the glycolysis will be happening. The polysaccharide will be break down and it will convert into the monosaccharide. So that monosaccharide will be accumulated into the liver as a glycogen. The excess of monosaccharide will convert it into the other matters, that is a fatty acids. Okay, so that is very important one. And gut brain crosstalk, and satiety. So satiety means that is a satisfaction. When your tummy is very full, that the signal will go to your brain. Okay, now our tummy is full, so there is no need to take more food. That is a satiety. And mood and pain, everything will be designed by, that is a hypothesis that was said by many researchers. That is regulation, that gene regulation is taken care by the gut microorganism. That's what here I'm mentioning, gut and brain crosstalk. Okay, that is the like roadmap of that is a cross roadmap between the gut and the brain. That is the satiety, mood, or whatever. This kind of signaling, neurotransmitter signaling will be will make in the gut region, and it will go to the brain. Then only we can say like there is enough food, there is no need for more food. That we can tell like a mood or pain. That is the same one that we are going to discuss today. And next one, gut microbiota, this is your health. So immunity, it is directly involving in the immunity, vitamin, and the metabolic pathway, and that is very important one. And indirectly it will involve in the obesity process, and definitely it will cause some inflammation. And very recently one of the scientists is find out that it is also involving in the autism process also. So this gut microbiota play a very important role in both beneficial and harmful ways. Next slide. Next slide. So the probiotic intake, the beneficial of this gut microorganism is, first one is SCFA, that is a short chain fatty acid production and antioxidant production. And indoxyl sulfate, that is a very important one. Indoxyl sulfate, it is not only kidney toxin, it is also a liver toxin. That is a tryptophan is involving in the indole production, then indole will be converted into indoxyl sulfate. It is reducing the indoxyl sulfate so that your kidney and liver will be benefited. So your gut microorganism will involving, it's like a god, that's what I'm telling. It's like it's extended his hand into the all the region, right? It will involving in the neurotransmitter of the brain, and it will involving in the production of protection of kidney and the liver, and improved lipid metabolism that we discuss in the later slides, and a low gut inflammation, insulin sensitive, and reduced risk of some infections. So these are all the matters is in your health aspects. It is a good to your health. The next one. So major functions. The first these gut microorganisms are involving in the 4 major functions. One is the metabolic role, that is directly involving in the digestion process or fermentation of non-digestible foods, and involving in the absorption of nutrients by intestinal cells. And the second one is barrier role, that is a shelter, that is a shield against microbes and toxins and production of protective mucus by the intestinal cell, and third one is the defensive mechanism. It is development of the intestinal immune cells, immune system, and fourth one is maintenance role. Here the maintenance role means the maturation of the gastrointestinal tract, maintenance of intestinal mucosa, production of mucus, enzymatic activity by the mucosa. So that is very important. That's what I said. The organism first it will reach into your mucosa. The mucus contain lot of enzymes. If the organism is very virulent, it can easily escape from the mucus activity, mucus, then it will reach into the mucous membrane. And the breast milk automatically, next slide. Go to next slide. So the breast milk contain a lot of things like minerals, vitamins, and the nutritional composition, hormones, growth factors, microbial communities, microRNAs, and everything. But one thing you should understand that, SIGA and lactoferrin, next one. SIGA and lactoferrin, it is directly involving in the degradation of, it is directly involving in the decreasing of Enterobacteriaceae family, and HMO, that is a human milk oligonucleotide, is involving in the increasing of bifidobacteria. And if no gut microbiota, if the gut microbiota, next one, if the gut microbiota will not be there, what will happen? What would happen? So just imagine if the gut microbiota directly involving in the macrophage activities, so that if the macrophage activation is not happen directly, that is the normal immune function will be disturbed, so that dysbiosis will happen. Necrolytic enterocolitis, that is a necrolytic enterocolitis, as you know very well, it is a devastating disease that affects mostly the intestine of premature infants. That is a very, 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 very important one. Directly it will affect the formula feed or whatever. So the organism will be evolved in your body. If the organism is not there, it is not activating your immune system. If the immune system is not active, definitely the opportunistic pathogen, the gut microorganism is also, some of the gut microorganisms are also called as opportunistic pathogen. So this one will easily affect your, causing some infection, and directly it will cause the necrolytic enterocolitis, and it is a directly affect your intestinal tract. So that's what here I am mentioning. And apart from that, some autoimmune disease will come, atopy and obesity may come. And the next one, gut microbiota versus food. If you are taking a different kind of foods, normally we are taking lot of food, high fiber diet or red meat or high fat diet or whatever. So whatever food you are taking, some of the microbial phylum will be there in the digestion process. Here you can see. So these are all, I'm listed the bacteria. That's what I'm telling. Akkermansia. Akkermansia is a very important one. Akkermansia is directly involving in them. It is a mucin degrading bacteria, and it is a very, very important one in the gut region. And mucinephilia has been inversely associated with the obesity, diabetes, inflammation, and metabolic disorders. And once these are all the organisms are involving in the degradation process, and after degradation, the long chain fatty acid and we will get LPS and cytokines, and the long chain fatty acid we will get it. And very important one, that is a short chain fatty acid, SCFA, and from that we will get acetate, pyruvate, butyrate, and propionate. And the second important one, when we are taking the red meat, normally it will take 14 hours for the digestion, and it will convert. It will converted into phosphatidylcholine derivatives. Okay, TMAO, and TMAO means trimethylamine N oxide. It is an important gut microbe dependent metabolite, and L-carnitine and choline will come. Okay, choline will come from, these are all the food matters. And very important one, next one. And microbiota in the intestine. Here, that's what I'm telling, very important one. Here you can see this one in the right side, that is the block adhesion. The block adhesion, the cell, that is a intestine. Just imagine this is the large intestine. Inside the large intestine, that is a mucous membrane will be there. So here, that is the structure of the Y shape is bifidobacterium, red color, and the blue color is a lactobacilli. Here you can see that is a prebiotic is nothing but HMO is a prebiotic. Just to take example, that will be accumulated on the receptor, that will be bind on the receptor of your epithelial cell, so that the receptor decoy will happen automatically. The pathogen will not easily attach on the receptor. So this one, it will do the block adhesion. And the very, very important one, this prebiotics will be converted into small monosaccharide, and it will be easily absorbed by the that's what I'm here I'm giving this details. Here, this is a very, very important one. That is, a gut microbiome has a regulatory function on the host energy metabolism. Next slide. Yeah, next one. This is a common one. Just I'm giving. This is the way the just mucous membrane, the microorganism, metabolites, and proteins and the genes will be entered into the mucous region. That is the intestinal region. Here you can see the mucous region. Just I'm giving this picture. The non-digested polysaccharide, is that is a fermentable or non-digested fermentable polysaccharide that will be converted into monosaccharide first. Then this will be entered through that cell, through the absorption. It is entered into the, it is easily penetrate into the cell. That is a active transport and passive transport. You already studied, you know, you are aware of it. And this, especially the microorganism, commensal microorganism, here that is orange color, the commensal microorganism is helping in the absorption process, so that that monosaccharide will enter into the cell and it will reach to the liver, and it will be accumulated as a glycogen. Then the excess amount of monosaccharide will involve in the lipogenesis, and it will be accumulated as a free fatty acid or accumulated as a lipoprotein. That is a lipoprotein. And then here you can see in one more side, that is divided into two, that is converted into two. One is a monosaccharide, another one is SCFA in the top region. The SCFA is a short chain fatty acid. Short chain fatty acid is divided, and it is attached with GPR 4143. That is, GPR is G coupled protein receptor, that will be bind with the protein receptor and it will enter. That's what they are. The color is different. Here they are mentioning enteroendocrine cell. Here this one is a very important one, and this receptor, this single chain short chain fatty acid will be attached with just GPR. This GPR is a coupled one, so that is a 41 and 43, and that is one is activated when the short SCFA will attached on this GPR 4143. It will be activated so that it will be directly entered into the epithelial cell through the absorption process by the help of bifidobacterium, and it will directly, these are all the organisms are involving in the human gene regulation. That's what here we are mentioning. Okay, so it will be reached to the PYY. PYY means it's nothing but peptide tyrosine tryptophan. The Y is a singlet representation, right? When we are talking about the amino acid, we are using the triplet, like tyrosine or whatever, you know, TYR we are mentioning. Glycine means GLY. Here we are mentioning about a PYY means Y is a singlet representation. It is a short peptide, that is a human, that human is enclosed by the PYY gene. It is released from cells in the ileum and colon in response to feeding. And the very, very important one is it is a neuropeptide and involving in the signaling process. So what will happen there? If it is released directly, it will inform to brain, that is involving the neurotransmitter, so that it is a signaling will be passed. So what will happen? The bifidobacterium FIAF, that is a angiopoietin like 4, that is a peptide one, that is a protein. FIA is also known as angiopoietin like 4 protein, and the function is it is a stop the lipolysis. If the lipolysis will be stopped here, that's what here mentioning, the FIAF will be there. The symbol, that symbol is a inhibition symbol here next to this one, you know, FIAF. That one is a inhibition symbol. If the inhibition, it the bifidobacterium involving in the through the gene regulation, if it will inhibit the FIAF, directly the lipolysis will not be happened. So if it will not be happened, we will not get the proper energy. So what will happen? This organism will directly inhibit the FIAS, so that one inhibition process of that lipolysis will not be happening, so that the lipoprotein will be converted into small fatty acid, free fatty acid, so that free excess fatty acid directly involving in the muscle formation, and it will deposited in the muscle, and excess amount of this free fatty acid will be deposited in our adipose tissue. And the adipose tissue will be normally the higher amount of free fatty acid will be accumulated. It will be accumulated. It will be through the leptin. Through the leptin, it will inform to the brain that is the satiety. That is a enough condition. There is no need for taking more food. That signal will transformed through this adipose tissue. Then through the adenonectin process, it will involving in the AMPK process, that is a activated protein kinase phosphorylated AMPK process. Again, it will converted into free fatty acid oxidation. That is a degradation of free fatty acid. Again, it will be deposited. Again, it will be converted into glycogen deposited in the liver. Otherwise, it will go to the muscle. Okay, muscle addition. So this is the process, especially with the help of microorganisms. This is the process is happening in our intestine. This is the very, very important one. So directly this gut microbiota, this gut microorganism is involving in the gene regulation process, so that it will be regulate the energy, and it will be easily involving in the neurotransmitter, as well as it is involving in our immune system. And it is also very important thing, through that probiotics, the organism is degrading the probiotics, and it will be that probiotic will be adhere on the receptor, so that the decoy process will be happening, and the mimic process is also happening with the help of this gut microorganism. So this is the entire process. Next one. This is the detailed matter. Here I am just giving some overview. Just these are all the bifidobacterium affect probiotics, and directly it involve the degradation, and it will block the pathogen in one way. On the another side, it is involving in the fermentation process. pH will be reduced, and it will suppress the pathogen entry, and it will suppress the pathogenic proliferation. This is the overall view. And the next one. And this is the functional microbiome approach. Here we are just taking the, so if you have any doubt, you can ask me. I have given my email ID so that you can send me any mails regarding this. I can share I will give the answer. The session is open for discussion. If you have any doubt, you may ask me. And some of my students are also working on the bifidobacteria and some antibiotic resistance studies. They are working on this one now. So, shall I wind up? Shall I wind up the session? Or any doubt regarding this? Yeah. So thank you so very much, especially Dr. Balasubramaniam and the team members who have given me an opportunity. Thank you, participants and students, patiently who have listening my presentation. So any doubts, sir? I have given my mail ID. It is also available in my institute website. So you can just drop a mail.
So there is a question in the chat box, sir.
Yeah. Please just open it.
How SCFA is useful to body?
Short chain fatty acid is directly just degraded by the microorganism, and it will be converted into, that's what I mentioned in that slide. The short chain fatty acid will be activated in the GPR 41 and 43, and it will be involving in the peptide YY, that is a peptide, and it will create the signaling, and directly it will be involving in the adipose tissue formation. And if the excess amount of adipose tissue will be there, again it will go to the conversion of free fatty acid. Again it will go to the involving, that is after the glycolysis pathway. Again it will go to the free fatty acid oxidation process, and it will be settled as a glycogen in the liver, or it will directly go to the muscle for the building block of your muscles. So any other matter?
Okay, sir, let us wind up the session. So thank you very much for your effective presentation, sir.
Thank you. Thank you all.
Thank you, sir. Dear participants, the next session will be started at 2 p.m. The participants of poster presentation are requested to be ready at that time. The next session will be started at 2 o'clock. The participants of poster presentation should be ready. Mr. Subrota Hati, you should be ready at that time. First poster presentation will be on anti-hypertensive peptides derived from fermented camel milk by proteolytic lactobacilli in silico in vitro study. And second poster will be in vitro study on purification and production of antioxidative peptides from fermented goat milk produced by Lactobacillus fermentum. And third poster will be reduction of blood sugar level depends on neurophytochemicals. And fourth one will be NUTRIGENOMICS. After completion of the first poster presentation, the oral presentations, the remaining oral presentation of previous day session will be discussed then. The participants of oral presentation also need to be ready after the completion of poster presentations. We request the participants to leave the session now. Once again, the session will start at 2 p.m. The participants, if you have any doubts, you can clarify it now. Participants, if you have any doubts regarding the presentation, poster presentation will be started first. First presentation will be on anti-hypersensitive peptides derived from fermented camel milk by proteolytic lactobacilli. Thank you, everyone.
Hello. Hello. Hello.
Good afternoon, everyone. We are going to begin the afternoon session. The presenters, be ready with your presentation. We are going to begin the next session. The presenters, be ready with your presentations.
Sir, can you tell me the name of the first presenter? Then I can keep my screen sharing ready. Hello? Hello?
Hello, good afternoon, ma'am.
Yes, sir. Can you tell me the name of the first presenter? I am the oral presenter, Dr. Arvinda Shaw.
Yes.
So
Okay,
my
ma'am.
position, can you tell me?
Yes, just wait, ma'am. Let the chairperson come to the session. After that, you can start your presentation. Please
Sir,
wait,
I
ma'am.
wanted to know, am I the first person to start? Then I'll keep my document ready for screen sharing.
You can present later, ma'am. First is poster presentation. Next, after that, you can present, ma'am. First is poster presentation, ma'am. Afterwards, you can present your paper.
Okay, okay. Thank you,
Thank
sir.
you. Thank you.
Okay.
Thank you, ma'am. Thank you.
Good afternoon, sir. Yes, I am ready, sir. I am poster presenter.
Good afternoon, sir.
Today is share my screen.
Good afternoon, sir.
Good afternoon.
Please wait, sir. Let the chairperson come. After that, you can start your presentation, sir. Please wait, sir.
Okay.
Okay, okay, sir.
Good afternoon, everyone. I hope you have found this morning presentation informative and useful. Thank you for spending time with us today. Now I take this opportunity to call upon Dr. B. Harinathan, Assistant Professor of our department, to introduce the chairperson.
Good afternoon, sir. I feel really happy to introduce Dr. K. Anandaraj, sir, who is serving as a principal, associate professor, and head in Shanmuga Industries Arts and Science College, Thiruvannamalai. He has got a very good research experience of about 19 years. His thrust areas of research are immunology, biotechnology, and medical microbiology. He has published around 15 research papers in indexed journals, and also he has published 3 book chapters. He is an active researcher who is also guiding 2 PhD scholars and 1 MPhil scholar. We are really proud to have you on this occasion, sir. Thank you.
Yes, thank you very much, sir. Thank you.
Thank you, sir. Thank you, sir. I request Sabrota Haiti to present your findings.
Okay, sir. I'm
Okay.
just sharing the slide. Kindly stop your presentation, sir, so that I can share my slide.
Okay, okay, sir.
I think you can see my slide.
Yeah, sir. It is your slide is visible, sir.
Yeah.
So you
Good afternoon, all.
kindly display in full screen, sir. Possible.
Full screen, then it will be because it is in vertical, na? So the letter will not be in a proper way.
Okay, sir. Okay, sir. Okay, sir.
Yeah. Good afternoon, all, and the respected judges and chairmen. I am going to present posters on anti-hypertensive peptides derived from the fermented camel milk by proteolytic lactobacilli in silico and in vitro study. Our aim is to all of you know that the camel milk has an anti-diabetic effect, and Amul is selling this product by claiming that they have a very good insulin-like protein materials are there. But we want to explore it another biofunctionality, that is the blood pressure control, means anti-hypertensive activity. So we are getting the camel milk from the Kutch area of Gujarat, where Amul has installed their plant. So we are getting the camel milk from the Kutch area. Then after getting the camel milk, means they are giving us the camel milk in the form of camel milk powder, and we are making it in milk in the reconstitution. And then we evaluated the anti-hypertensive activity of this camel milk by following in vitro studies. So for this study, we have decided 4 objectives. First is to evaluate the PEPEX and S inhibitory activity of the lactic cultures, then to optimize the growth condition for the production of peptides. And we have also optimize the relative proteolytic activity of the cultures, and then we have purified and characterized some novel S inhibitory antihypertensive peptides from the fermented camel milk. So that was our objectives, and for this study we have estimated PEPEX activity, S inhibitory activity, and also we have measured the peptide content using OPA method, and we also measured the proteolytic activity by HPLC through relative proteolytic activity. To have an antihypertensive activity, the camel milk peptide must have a smaller fragment of peptides. We have also estimated di and tripeptide activity, and then we have also see the peptide pattern by using the RP-HPLC method, and also we have done the sequencing through LC-MSMS, and we have seen the protein profile through SDS page, and also purified the peptides by using the 2D gel electrophoresis. So here is the result. So here you can see that the PEPEX activity, we are getting the PEPEX activity for different cultures at different amount, and we have seen that the NS4 has given the best results, and then S inhibitory activity is also getting higher, and also we have estimated the proteolytic activity. You can see the proteolysis profile by with the help of the sonicator, and then collect the materials and then added in the particular substrate and see the activity. We have also measured the antihypertensive activity. It is called S inhibitory angiotensin converting enzyme inhibition activity, and we have seen for different fragment of the peptides. This is a 3 kDa fragments, and this is 10 kDa. So we have passed through the water soluble extract, means the peptides by passing through different ultrafiltration cut-off membrane, and we have seen that the lower peptides are giving better results. And we have also measured the peptide content of those 3 kDa and for 10 kDa permeate and retented, or even 5 kDa permeate and retented samples. Then we have estimated these peptides in the HPLC. So compared to the camel milk itself, you can see these peptides are getting fragmented, and the peptide profile, means the peptidomic profile, are increasing as you are fermenting the camel milk. And then we have see the protein profile of those peptides. So we did the SDS page. So this is a ladder, and this is the control sample. These are two control samples, and these are the different bacteria we have used for the fermentation of the camel milk. And you can see very clearly from this picture that the hydrolysis was taking place in the higher molecular. So the lower molecular weight peptides are also getting 1 kilodalton and 3 kilodalton peptides we are getting. So we have purified these peptides through the 2D gel electrophoresis. These are the different 2D spots. And after collecting these 2D spots, we have done the trypsin digestion. After trypsin digestion, we have done the LC-MSMS. After doing LC-MSMS, the raw masses which we got through the LC-MSS, that has been analyzed in different online databases. This is called BioPep, then PDB protein databases, then PIR protein information databases, then HTDB anti-hypertensive databases, and we have got 3 very good novel peptides: GPP, YQ, PL, VPR, and CISS, SSS, TPP, YDL, NRK, FK, and VCNY, VSWIK, and MDTI, EP, VS, BS, CIS. So these 4 peptides we have got it after purification and characterization from the camel milk. And these 2 cultures, NS4 and 09, NS4 is the Lactobacillus rhamnosus and 09 is the Lactobacillus acidophilus. So these 2 organisms have given these noble peptides. It is the anti-hypertensive peptides, and we have identified these peptides by using this entire method, and we have deposited these peptides in the NCBI site. So the nobility of the work is that peptides first time from the Indian breed, the camel from the Indian origin, we have deposited in the NCBI site, and now people can use the reference data from our resources. So that is the nobility of the product, and we are going to produce a acinibutary rich peptide-rich fermented camel milk because we are working on it by enriching these peptides. So that is our main motto, to produce a fermented camel milk like lassi, which having these anti-hypertensive peptides. So thank you all. That was my aim, and that is our work, to produce and purify, characterize the anti-hypertensive peptides from the camel milk using our own lactobacillus cultures. Thank you, sir.
Thank you for your informative presentation, sir. Now the session is open for discussion. Nagraj sir, if you have any queries, you can ask, sir.
Yes, sir. Please ask him to present the page, sir.
Yeah. I should present the PPT. I should say PPT.
Sir, present your presentation. That is that slide.
Okay. Just I'm going back. Yes, sir. I'll bring my slide, sir.
Yes, sir. Please.
Can you see my slide?
Yes. I'm able to see your slide.
Yes, sir. Yes.
Yeah.
So, my first question is, why you choose this camel milk, sir, for this research work?
Because, sir, camel milk we have seen, because we want to explore the camel milk to make a good product out of it, sir. Because mostly the camel milk has a very good, commercial values, and people are selling it at a higher amount. So our aim is to, because we are very close to the Amul, we are here in Anand. So Amul means the federation, jo Amul companies hai, they have given the responsibility to us to make a good product of by using camel milk. They are supplying camel milk to us, and they said that you explore another biofunctionality if possible, so that we can claim the things that anti-diabetes is already there, so we can also have another benefits of anti-hypertensive, so people can consume it, so we can have a more health benefits and we can sell it more.
Okay, very good presentation, please.
Thank you, sir.
Yes, sir. Go ahead, sir. Next.
Okay, sir. Okay.
So should I stop here, sir?
Yes, sir. Please close it,
Yes,
sir.
sir. Yes, sir. Thank you. Thank you, sir. Thank you.
Oh, okay, sir.
Gaurav Kumar Panchal, are you here, sir? Annapurni Sivanandam, are you here? Annapurni, are you here? Dr. Aravind Shah?
I am here, sir. Should
Yes,
I start?
Can you present
Yes.
your findings, ma'am?
Yes, surely.
Okay, thank you.
I'm doing the screen presentation, sir, now.
Yeah, okay.
Sir, is my screen visible?
Yeah, it's visible. Please go ahead.
Okay, sir. Okay, sir. Hello, everyone. Very good afternoon. My regards to the chairperson, and thank you to the organizers giving me this opportunity. Myself, Dr. Arvinda Shaw, Assistant Professor, Department of Food and Nutrition, Raidigi College. And today I'm here to give some simple information on sickle cell anemia, which is a preventable blood disorder. But the time has come that we should spread awareness regarding this disease. So the introduction is that sickle cell disease is a group of inherited disorder that affects the hemoglobin, which is primarily a protein in the red blood cell, a pigment that is responsible for carrying the oxygen to the tissues. The most common type of sickle cell disease that can be seen among the Indian population is sickle cell anemia. Now what happens, our red blood cells are usually round in shape, and so they are flexible and they can move very freely to the blood vessels of our body, and without causing any clot or obstruction while their movement in the circulatory system. But what happens, anyone who is having the sickle cell trait or the gene, due to mutation, what happens, the hemoglobin gets converted or mutate to hemoglobin S, which is the abnormal hemoglobin. Now the regular hemoglobin gets replaced, that is hemoglobin A gets replaced by the hemoglobin S in such kind of people who carry the sickle cell genes. And thus what happens, the normal shape of the red blood cell, which is supposed to be round and flexible, gets changed and it gets morphed into crescent shape or moon shaped. What happens when the sickle cell shaped RBCs form in the blood circulation? They are irregular in their shape. As a result, they get clustered together, and being unable to move, as the result, they get stuck in the blood vessels, arteries and capillaries, and they hinder the supply of the oxygen to different part of the blood. Since sickle cell cells are not flexible and they cannot change their shape according to the route of the circulatory system, many times they get clogged and gathered and they burst while moving through the blood vessels. Sickle cells have a lifespan of 10 to 20 days only, whereas a normal blood cell, RBC, has a lifespan of 90 to 20 days. So it is very common that these sickle cells, since they expire in 10 to 20 days, these people turn anemic. This causes rapid loss in our red blood cells, and thus what happens, it is not possible to recuperate so fast, and these people develop a condition called sickle cell anemia. Now, what are the complications that are related with sickle cell disease? Now here, since the sickle cell gets stuck everywhere while traveling, they cause a delay in supply of oxygenated blood to different parts of the body. And at that time, there are painful episodes, which is known as crisis periods, since the different parts do not get. It leads to organ damage, specifically spleen, kidney, lungs, brain. And anemia comes with its own sets of problems like fatigueness, shortness of breath, delayed less growth in children, development, and complication. High blood pressure is also seen among 33% of the sickle cell anemia subjects, primarily children. Now, what is the prevalence rate? It has been seen that 5,200 live births each year in India are from sickle cell anemia. That is almost 5,200 lives births that takes place. The newborns, neonates are suffering from sickle cell anemia, and India has the second highest burden of this disease, apart from sub-African countries and many of the Middle East countries. So various scientific studies that have been conducted throughout our country, it has been found that this sickle cell anemia, which is a blood genetic disorder, is widely prevalent among the scheduled caste groups and the tribal people. So it has been seen, different studies by ICMR has been seen that almost 1 to 40% of the tribal population in India, mainly locating in central and southern India, they are suffering from this sickle cell anemia. And it is estimated that 1,800 million Indians with sickle cell trait, that means if they get married to someone else who is also having the sickle cell trait, then their newborn will become a definite sickle cell anemia patient. And 1.4 million people, tribal population, are there who are suffering with this sickle cell disease. So it is very unfortunate that such a large number of children are still being born in our country who are suffering from these genetic disorders, which is completely preventable through awareness. So what is the treatment? The treatment involves plain relief medication, hydroxyurea, antibiotics, childhood immunization, blood transfusion, bone marrow transplant. But many of these things are very risky and it might not give the proper solution. So regular medical checkups, health care, change in healthy lifestyle, and avoidance of situation leading to pain crisis should be avoided. So what is the prevention method that can be done? The most important part is screening of the people. Even the educated and the uneducated people are not aware whether they are carrying the sickle cell trait in them or not. Meticulous screening and the spread of awareness among the community people, especially the tribal people, who are socio-economically disadvantaged and they are medically under screened. So it is best that simply prevent the birth of affected children by avoidance of marriage between trait people. Trait people means, I have given an example. Suppose a male has a trait, sickle cell gene trait, and a female also has a sickle cell gene trait. If they go to any of the screening center before getting married, and if they find out that they are carrying the sickle cell trait, then they should never get married. And if they get married, then they should refrain from getting their own children, because there is many chances, because two sickle cell trait people, if they get married, then their offspring will definitely suffer from any one type of the sickle cell disease. So prenatal diagnosis is one of the most important aspects for program. Even if these people get married, once the cases are identified, counseling plays a very important role, providing supportive measures like use of oral rehydration solution, drinking of a lot of water, avoiding climatic condition, and complete immunization, prophylactic dose of penicillin vaccine, and other things. Therefore, the successful prevention program is good peripheral units that is required so that the screening can be done for all people, whether tribal or non-tribal, whether in rural areas or urban areas. So in conclusion, we can say that it is a well-known fact that sickle cell gene is widely prevalent in tribal population of India, and Indian Council of Medical Research and National Rural Health Mission, they have undertaken many outreach programs in different parts of the country. But still, Many people, even the educated people, are not aware that early detection of sickle cell disease is extremely important for improving the quality of life for these people. Awareness about the disease should be generated through mass media and health organizations also, because this is very much preventable, and we should make sure that our future citizens should not suffer from this kind of disease. Thank you so much. Thank you, sir. Hello? Hello?
Yes, thank you. Thank you, ma'am.
Okay.
Now it's time for discussion.
Yeah, Aransha, ma'am.
Yes, sir. I'm there.
Ma'am, I have one question, ma'am.
Yes, sir.
Why this SCD is occurring only in the tribal? That is mostly high percentage of tribals are affected with this SCD.
Actually, sir.
Which factor?
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir. Actually, sir, I have gone through many of the scientific papers, and it has been shown that the data is actually undocumented. Many of the studies that have been performed in the central and southern part of India related to the tribal people, because they are socially, economically underprivileged and the medical facility is not available. And it is seen in them that the people of same trait, the trait is there also, and they are getting married among themselves, and as a result, when they are having offsprings, most of the children are suffering. I think even in cities, if we get this kind of screening, then definitely the scientific studies will come up with people in well-to-do families also, and other families also, with these kind of things. Maybe they are lucky enough that the two gene people are not getting married. Maybe someone is carrying the trait and the other person is not, so the symptoms is not coming up. But if proper studies carried out in different part of the country, not alone the tribal part, then I think other studies will also make sure that this is widely prevalent. At least the sickle cell gene is definitely prevalent. The screening is not done, maybe because of awareness. People don't be in India, we more prefer before marriage a horoscope match rather than a blood test match, or this kind of a sickle trait or a thalassemia match, which should be generated with awareness. That horoscope is definitely important, but this kind of matching of the traits or the other kind of disorder is not there in the partner is also very important to make sure, because the newborn, the offspring who has never done any kind of a mistake, he will suffer throughout his life, because the quality of the life gets compromised with these kind of situation and condition, because there's no cure till now. So it is all through the prevention and taking care of him, regular health care checkup, that the child has to survive. And in many cases it is seen that these kind of child who is a hardcore sickle cell anemic, they hardly stand to live till 15 years of age. And if they do, then they survive with so much of complications, regular blood change, giving them blood because they are anemic. They are not able to participate in any kind of activities that requires a lot of stress, strain. So they move out of the mainstream. They just can't live a normal, healthy life, which is just not which is not desirable. As a parent, we should always make sure that our offspring gets a healthy life. That is his requirement, and that is his should be the priority for the adult people, to make sure whether any one of them are having this kind of genetic disorders. or the genetic mutated genes in them, then they should make sure that the spouse, at least their spouse, should not have that. Because if two people having the same kind of genetic trait, if they have, then it is doubly sure that the child will born with this kind of sickle cell disease. It can be thalassemia also, it can be sickle cell anemia also, or other variants also.
Very good, ma'am.
Thank you, sir.
Is there any research work in progress related to gene therapy to prevent this SCD?
Sir, there is no such cure. What Indian government, especially in different kind of tribal locations, I had gone through a study that is being taken in Gujarat. What they are doing, sir, when they are doing this kind of screening camps, they are doing in especially socio-economically disadvantaged areas, among the tribal and the non-tribal areas. What they are doing, they are arranging for camps, and there they are getting the simple blood test, special blood test called the sickling test on RBC. And HPLC, this kind of different kind of tests they are doing to track people who are having the sickle cell trait in them. And then they are counseling them about the prenatal diagnosis or about their marriage, that you should not, see, you are having this trait, you should not get married to someone. And there they are providing them typical kind of a card, because these terms might not be applicable for those people, they are illiterate. So they are providing them yellow colored card with 3 parts. If they're having the gene trait, then one part of the card is yellow. If the partner is also having the gene, then they have both the parts are yellow. So some kind of a color identification cards are being given, which is making them aware, see, you are having something, and if you want a healthy child, then you should make sure that you should not get married to someone who is also having the same colored card, because then your child will born with this kind of inherited disease. So this kind of different kind of initiative is taken by ICMR and other local rural camps, but it is still, the awareness is not there on the social media as well as mass media. We can hardly see anything about sickle cell anemia. Everybody knows about anemia. We should eat iron rich food and this and that. We should eat citrus fruits because vitamin C is important for absorption of iron, but we hardly see any kind of advertisement on the national level or other kind of platforms like social media where people discuss about this sickle cell anemia or thalassemia, which is very much preventable if the people who are carrying the trait don't get married to someone who is also carrying the trait, because in double hemoglobin SS, that means the child will suffer from this sickle cell disease. So this is some of the things that can be prevented only through awareness and educating people, whether in tribal area, non-tribal area, urban area, or villages.
Thank you for your informative and valuable presentation. Sorry to interrupt you because we are already running short of time, ma'am.
Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Okay.
Thank you. Thank you for your useful presentation, ma'am. Thank you.
No, okay, sir. I can understand. Thank you, sir.
Thank you. Thank you.
Ms. Jahanvi, are you here?
Yes, ma'am.
You can start your presentation now.
Okay, ma'am. Ma'am, can you see my presentation?
Yes.
May I start, ma'am?
Okay, you'll be given 3 minutes for your presentation and 2 minutes for the discussion.
Okay, no problem, ma'am.
You need not concentrate on the introduction. You can straightly go for your materials and methodology, and you can go for the results then.
Okay, no problem, ma'am. Okay. Good afternoon, one and all present here. My name is Janvi, and I'm here to present my study on evaluation of in vitro cholesterol removal ability of potential probiotic organism. So let's see directly how the probiotic organism is able to remove cholesterol. So it is able to produce the bile salt hydrolase enzyme, which is very useful while cholesterol removal. Then it can also assimilate the cholesterol. It can also directly bind the cholesterol molecule to the bacterial cell walls, and also the physiological actions of end products of short chain fatty acid fermentations. So here in this study, I have first determined BSH activity, that is bile salt hydrolase activity of our probiotic isolates with plate assay method. Then the potential BSH producers were taken for further study, that is cholesterol removal, and this cholesterol removal was studied by orthophthalaldehyde spectrophotometric method. The culture which showed the highest cholesterol removal was taken for further study, and the cholesterol removal with time profile was studied. Then the different concentration of cholesterol was studied for removal. Then after live and heat kill cells, that is dead cells, were studied for cholesterol removal ability. At last, the determination of cholesterol removal from cholesterol rich food like egg yolk with the simulation treatment, that is gastrointestinal simulation, was determined, and the potential isolate was lastly identified by 16S rRNA sequencing method. So here in the result, I would like to brief introduction about the earlier work. That is, probiotic organisms were isolated from different sources such as fermented curd, batter, lentils, human milk, and fecal sample of baby. Then total 27 different isolates were achieved on MRS agar plate, and they were primarily screened out with acid and bile tolerance, and I got 12 different isolates. Then these isolates were further screened by simulation treatment, that is gastric, intestinal, and gastrointestinal simulation, and 9 potential isolates were selected for further study. In this study, one important characteristic, that is cholesterol removal, was determined, where BSH activity was studied by plate assay method, and out of 9, 5 isolates showed a zone of clearance. As we can see in this picture, when MRS plate was supplemented with ox bile and also CaCO3, the zone of clearance by producing the bile salt hydrolase enzyme, which is able to utilize the bile. I have selected 3 different culture, B11, D25, and B32, and further cholesterol removal study was taken. First of all, in this study, only 200 microgram per ml concentration of cholesterol was given, and cholesterol was given as a solely source of nutrient in the medium. No other nutrients were given to the organism. So after 24 hours, the isolate B11 showed maximum cholesterol removal percentage, that is 87.7%. Followed by 69.5 and 64% by B32 and D25, and also the growth was maximum in B11 after 24 hours. So B11 was selected for further study. When cholesterol removal was studied with time profile about 2, 4, 6, 12 and 24 hours, again we can see that in only 6 hours it showed 61.24% of removal, and after 24 hours 88.17, hence 24-hour result was more significant, but still it gives very significant result in 6 hours. When this cholesterol concentration was increased up to 500 microgram per ml, culture was able to show 72.55% removal till 500 microgram. As the concentration was increasing, also the cholesterol removal was decreasing, but still at 500 microgram, the high concentration, we also get up to 70% removal. When live and heat kill cells were studied, so at that time with dead cells, only 8 to 9% removal was achieved. Maybe this is due to the direct binding of cholesterol molecule to the dead cell wall of the probiotic organism. So it showed very negligible amount of removal, but live cells were able to remove cholesterol very significantly. Then I have tried it for cholesterol rich food, and I have taken the egg yolk. Egg yolk was homogenized, and the simulation treatment, that is a treatment with gastric juice, intestinal juice, and gastrointestinal juice, that is for 6 hours treatment, was given to this egg yolk. And after gastric simulation, that is 2 hours, 15.28% removal was achieved. After 6 hours of full gastrointestinal simulation, 45.09% removal was achieved. And when this flask was kept until next day, that is 24-hour incubation, we could observe 51.76% cholesterol removal by this
Can we
C11.
please conclude
So
your
it's a
research?
really significant. This is the last slide, ma'am. So this was identified by 16 as RNA sequencing method, and was identified as Pediococcus pentosecius, which is a very significant probiotic organism. So here I can conclude that if we are taking probiotic organism as a food supplement, it's really beneficial for there are so many health benefits of probiotics, but one of them is this cholesterol removal. And with my culture, I got up to 70% to 80% removal within 24 hours. So, thank you so much. Any questions?
Anandrat, sir?
Yes, ma'am. Please go ahead, ma'am. Go to next candidate.
Next
ma'am.
presentation. Okay, sir.
Yes, ma'am.
Okay, thank you, sir.
Mr. Karlajan, please start your presentation, sir.
Yes, madam. Now I can continue with my presentation. Let me share with the slide right now. Madam, there is a challenge in sharing with my slide. Could you please guide me? Hello?
Yes, sir.
There is a challenge in
Are
sharing
you
with my
in
slide.
meeting
What I
screen,
should
sir?
do?
Are you in meeting screen?
Yes, madam. Yes,
On your
madam.
right.
Yes, madam. It's so.
On your right, are you using laptop or mobile phone?
I'm using my laptop, madam. I'm
Which
using my
device are you
laptop.
using, sir? Laptop or mobile phone?
Laptop.
Laptop. So on your right, you can see options like present now. On your right.
Yes, I clicked it.
Yes.
Yes, I clicked.
After clicking that, you have to open your PowerPoint.
Okay.
Keep it
Okay,
open.
madam.
And then click the present now option. Select entire screen. Then go for the PowerPoint. It will be visible. Click the present now option. Select entire screen. In the taskbar, your PowerPoint
Yes.
already will be in open. Go for that. Then it will be visible for others.
Yes, madam. Madam, are you able to see my slide?
No, sir. You are not presenting now.
Okay.
Click the present now option.
Madam, entire screen? I have already clicked on present now. Hello. Madam, are you able to see my slides?
No, sir. No,
Oh, sorry.
it is not presented.
Sir, I opened my slides, then I go to present now. I clicked on present now, but it's not showing anything.
Vijayalalitha ma'am.
No problem, sir. Shall we move to the next presentation, sir?
Ma'am, I get him to present.
Sir, I can present next.
Okay.
Oh, okay, sir. Okay. One thing you can do. Can you send your slides to us?
Yes,
You
madam.
send
Yes,
a
madam,
mail. You send a mail to us. You upload your
Yes,
PowerPoint
madam. I will
and send a mail
it
to
now.
us.
Okay. Yes, madam. I will do it right away. Yes, madam.
Okay.
So that
You
I can
just send
present
us a
next.
mail. You just send us a mail. After this presentation, we will call you again, sir.
Okay, madam. Thank you. Thank you very much.
Really. Mukul Raj Kannan, are you ready? Are you ready with your presentation?
Yes, ma'am, ready, ma'am.
Start presenting now.
Ma'am, is my presentation is this way?
Yes, you are visible. Yes, your slides are visible.
Hello, all. I am Mukul Raj Kannan. I am from VS National College, Tamil Nadu. My topic is AI, Food Safety and Standard Authority of India. Let's see what is FSSAI. The Food Safety and Standard Authority of India has been established under the Food Safety and Standards in 2006, which consolidates various standards that have handled food-related issues in various industries and departments. FSSAI is formed under the FSS Act in 2006. It is established in 2011, August 5th. The headquarters of FSSAI is in New Delhi. The CEO of FSSAI is Arun Singhal. That's the functions of FSSAI. Food control is the regulation of the food supply, industry and performance of food laws by national or local authorities. The main purpose is to provide consumer that all products during production, handling, storage, processing and distribution are safe, wholesome and fit for humans for consumption. Let's see what is safety. Safety refers to handling, preparing, storing the food in a way to reduce the risk of individuals becoming sick from foodborne illness. Let's see what is FSSAI. FSSAI is responsible for the protecting public health and promoting through regulation and supervision of safe. FSSAI is made for the adoption of the science-based standards for food products and their production, storage, distribution, sale, and import regulate to ensure the availability of safe and wholesome food to humans. As per FSS Act, let's see what is the definition of food. Food, as defined in FSSAI Act, is any substance, whether processed, partially processed or unprocessed, which is intended for human consumption. What are the general requirements of a food business operator or distributor? the display of licenses, notices, certificates, etc., knowledge of handling practices, regular health status of food handlers, food safety messages depending on type of food being sold, special instruction on product food being sold is needed. Let's see what are the requirements for FSSAI license by FSSAI. As part of the CI license, of the following things be mentioned on packaged food labels and must check. First, they check the nutritional value, they check the expiry date, they also check vegetarian or non-vegetarian, and the FSSAI logo with the license number. Let's see what is food hazard. Biological, chemical, or physical agents in food with the potential to cause adverse health effects are called food hazards. It may cause during the harvesting, formulation, and processing, packaging, and labeling, transportation, storage, preparation, and serving. Let's see what are the three types of hazards. The three types of physical hazard, chemical hazard, and biological hazard. Physical hazard. Hazards due to the addition of any foreign object that may be intentionally or unintentionally in the food. It is called the physical hazard. They are broken pieces from crockery, small bones on shell left in the food, long and dirty nails, handling of food without wearing hairnet and hand gloves. Some of the examples are jewelry, addition of dust, metal pieces, plastic, films, or foils. These are physical hazards. And let's see what is chemical hazard. Chemical hazards are caused due to any chemical which may be naturally occurring or process-induced substances that may cause foodborne illness. These are called the chemical hazards. The hazard cleansing products and sanitary, insecticides, rodenticides, food coloring agents, equipment lubricants, chemical food additives, and preservatives which may cause foodborne illness. These are chemical hazards. And biological hazard. Biological hazards occur when food gets contaminated by microorganisms formed in the air, food, water, soil, animals, and human body. Microorganisms are associated into bacteria, fungi, yeast. These are the biological hazards. Finally, we came to the climax part. I thank you all for who all kindly listen to me, and I would personally like to conclude my topic by saying it in mother tongue. Nandri Vanakkam. Thank you. Thank you, ma'am. Ma'am?
Yes, thank you. Thank you for your presentation.
Thank you.
Mr. Kali Rajan?
Yes, madam, I'm here.
Yes. Are you ready, sir? We will present your slides.
Yes, madam. Yes, madam. You can present my slides right now.
Okay.
Yes,
Just wait
madam,
for a
please.
few minutes. We will present it.
Yes. Yes, madam, I can see the slides. Madam, can I present now?
Start your presentation, sir.
Yeah, thank you very much. This is Dr. Kali Rajan from Mulungushi University. The topic for my presentation is antimicrobial and antioxidant activity of secondary metabolites from the marine bacteria Vergibacillus species associated with the sponge Gallispongia diffusa. Gallispongia diffusa. Madam, next slide, please. Madam, next slide. Madam, next slide. Hello.
Yes, sir. Wait a minute, sir. Wait a minute.
Yes. So, yes, madam. Secondary metabolites. Secondary metabolites are the chemical substance that are produced by the bacteria and plants and fungi. In this research, we have targeted on the metabolites produced by the bacteria that is associated with the marine sponge Gallispongia diffusa. These secondary metabolites are not utilized by the bacteria for its development and reproduction and other purpose. These secondary metabolites are highly used for the treatment of lot of infectious diseases, and it is used to check its activity against cancer and other diseases. So the objectives of the study is isolation and molecular characterization of bacteria associated with the marine sponge Gallispongia diffusa, and then bioprospecting of sponge associated bacterial strain for the production of secondary metabolites using fermentation process. Then the third objective is evaluation of antimicrobial activity of the secondary metabolites against the clinical fishbone and biofilm producing microorganisms. Then the fourth objective is evaluation of in vitro antioxidant activity of the secondary metabolites. Madam, next slide, please. In this research, the sponge samples were collected from Gulf of Mannar region, Tuticorin coastal region, and then the sponges were identified by the scientist P.A. Thomas from Kerala. Then the next one is isolation of bacteria. Isolation of bacteria from the marine sponges. After the identification of sponges, the sponges were subjected to the isolation of sponge associated microorganisms. So the sponge samples were rinsed with artificial seawater in the laboratory. Then the sponges were cut it into small pieces and they were ground with the seawater using mortar and pestle, and they were subjected to serial dilution technique. Then after serial dilution, we have isolated various bacterial strains. These designated strains are CSD 1, 2, 3, 4, and 5. From the designated strains, we have isolated one strains. We have taken into one strain that showed good antagonistic activity against various clinical pathogens. Based on that, we identified one of the strains, designated strains as Virgibacillus species based on sequencing. Then that particular strain was subjected to fermentation, fermentation process using soapful marine broth. We used 20 liters of soapful marine broth for the fermentation process, and we got 20 gram of crude extract from the bacterial fermentation. Then the bacterial crude extract was subjected to antimicrobial activity against clinical pathogens, fishbone pathogens, and biofilm producing pathogens. Then we have studied the biochemical profile of the crude metabolite that is separated from the fermentation process using the strain Virgibacillus species. Then we have tested the crude extract against free radicals, whether it has a good free radical scavenging activity or not. So the madam, previous slide. Can you please show me the previous slide? Antimicrobial activity of secondary metabolites. So this table depicts the antimicrobial activity of the crude bacterial metabolite collected from the bacteria Virgibacillus species. You can see various bacterial strains mentioned in the column, and these are the various antimicrobial activity recorded against various bacterial strains. These are the and results for antimicrobial activity of metabolites produced by Virgibacillus species. These are the biochemical, biofilm producing bacteria. Then this is the biochemical analysis report of the crude metabolite. Madam, previous slides. Just show me the previous slide. Previous slides. So so alkaloids were present in the the crude metabolite, and it was negative for flavonoids, steroids, and and tannins, and it was tested positive for terpenoids, anthraquinones, and alkaloids. Next slide, please. The metabolites isolated from the bacteria Virgibacillus was tested for its antioxidant activity. This is the results of antioxidant activity of crude metabolite produced from the marine bacteria Virgibacillus species. It showed good DPPH scavenging activity when it was tested using 1,600 μg concentration. The activity was just 39.61. Then it was tested with the concentration 2000. The activity was recorded 46, and ascorbic acid was used as a reference sample. Next slide, please. Next slide, please. This table depicts the previous slide. Previous one. Yeah. This is the antioxidant potential of crude metabolite against hydroxyl radical scavenging activity. The hydroxyl radical scavenging activity was 60.77 when it was tested with the concentration 2000 μg, and the activity was dose dependent. When we increase the doses of the concentration, the activity was increasing, and you can see the standard activity produced by the ascorbic acid. Next slide, please. Next slide. This is the activity of secondary metabolites against the scavenging of nitric oxide free radicals. Here the activity was recorded very low. When which was tested with the concentration 2000 μg, the activity was just 21.83. It was very low compared to TPPH free radical scavenging activity and hydroxyl radical scavenging activity. Next slide, please. Next slide, please. Next slide, please. This is the ferric reducing antioxidant activity of the secondary metabolites. Here the metabolites showed the activity 208.76. This is the ferric radical antioxidant activity of the secondary metabolites. Next slide, please. Thank you, madam. Thank you very much for the help.
Thank you very much, sir.
Yes, madam. Thank you. Huh?
Krishna Kumar.
Krishna Kumar, are you ready with your presentation?
Yeah, ma'am. I'm sharing.
Start presentation.
Is my slide visible now?
Yes.
Yes, okay. Good afternoon, everyone. My topic is gut microbiome and depression. Depression is the most searched word of the last year, 2020, because most of the people were in their houses, most of them were lost their jobs because of the lockdown, and so that's the most, yes, depression. Major depressive disorder is its clinical name. It is one of the leading cause of disability among worldwide, and it is expected to be outranked heart diseases as
number one disease burden by 2030. Because of every year, more than 264 million people are affected. And in last year alone, 8 million people were dead because of suicide it caused by depression. So depression is, there's a big difference between depression and grief. Grief is just a loss of the loved ones and something we lost, that's it. But depression is something more. It has some clinical reason. It involves more than just the reason. It involves our gut microbes, the stress cause, experience, anxiety, a lot of things. So the depression causes, it is not, it causes just loss of focus and everything. A major depressive episode is defined as depressed mood or loss of interest or pleasure in activities for at least 2 weeks with at least 5 symptoms that disrupt important areas of daily life. So it is a survey that took in lockdown. 43% of Indians were suffering from depression, all because of the chaos that caused by the coronavirus. And the causes of depression. There are a lot of reasons for it. First one is the medical conditions. People have a fixed mindset that they are healthy, but when they came to infect by any disease or a disorder, they got depressed. Some medical conditions can trigger depression, including chronic pain, hyperthyroidism, diabetes, and Parkinson's disease. And medications such as corticosteroids can also able to cause depression, and some depressions are hereditarily passed through generations. Some serious illness and substance abuse can cause depression. So the next is symptoms of depression. First is depressed mood. Lack interest in everything we do, and we don't like anything at all. So anhedonia is the lack of pleasure by doing things, and excessive guilt, suicidal ideation. It is a very important stuff that going on within depression, because people with depressed are most commonly have the idea of suicide. So that is also a symptom. Changes in appetite, we lost changes in appetite, sleep, psychomotor retardation, poor concentration, and fatigue. So the gut microbes. Now we are getting into the topic. Gut microbiome is evolved by the birth. The birth is of two types. First one is the natural birth. In that the baby was taken through the vagina. But at that time, the baby get the microbiome from their mother. They get the lactobacillus, bifidobacterium, and other microbes that it gets through the vaginal microbiota. But when it came through cesarean, it just get the clean microbiota like Clostridium and the Streptococcus, etc. These may cause infections to the baby by birth. So the gastrointestinal microbiome is a diverse consortium of bacteria, archaea, fungi, protozoa, and viruses. Studies in humans and other mammals have implicated that microbiome in a range of physiologic processes that are vital host health, including energy homeostasis. The microbes that can also digest the food products like dietary fibers that give short chain fatty acids, which is an important factor for adipose tissue and other things. And it also helps in immunologic activity, neurobehavioral development. The shift in gut microbiota composition may contribute to a shift in the regulation of the host physiology. The number of microorganisms inhabiting has been estimated to exceed 10, which is 10 times more than the normal cells of the human brain axis. This is the pathway for connecting the brain and the gut. The brain axis is the biochemical signaling that takes place between the gastrointestinal tract and the central nervous system. Central nervous system, neuroendocrine and neuroimmunologic system, enteric nervous system, the vagus nerve, and the gut microbiota. So in this picture, it is the healthy and depressed, how it differs. Healthy brain has a normal adrenocorticoid hormone and reduced amount of cortisol, and homeostasis were performed equally, and immune system were at rest. Macrophages are doing their jobs, nothing overdosed. And microbiota, it's just a normal microbiota like lactobacillus and other bacteria. But in depressed, cortisol levels are increased, adenoid corticotropic hormones levels are increased, and there's a disruption in homeostasis. And the that dysbiosis causes microbes to produce inflammatory cytokines, which causes inflammation in various organs like large intestine, which may cause diarrhea and a lot of which affects the brain, which is the leading cause for depression. Microbes important. People with higher levels of coprococcus and fecal bacterium typically do better ratings, quality of life. Both break dietary fibers to produce an anti-inflammatory compound called butyrate. Coprococcus and dialyster are consistently depleted in people with depression. Germ-free rodents develop depressive-like behaviors following fecal transplantation, but not from healthy people. So the new important factor in this gut-brain axis, E. coli bacteria can produce serotonin, which is a happy chemical. It makes us happy, focused, and with life. And lactobacillus and bifidobacterium are GABA producing organisms. GABA, which is nothing but gamma butyric acid, is also it is a precursor for the SCA, short chain fatty acids. And dopamine, it is also a molecule which makes us happy. It also produced by E. coli, and acetylcholine, then noradrenaline was produced by lactobacillus. So pathophysiology and treatment of depression. So it is a paradox, which is depression is. It is we didn't particularly know which causes depression, gut microbiota or imbalance in the neurotransmitters. So when there is a mood oscillation, there is a change in the gut microbiota, and when there is an imbalance in gut microbiota, it also causes depression. So in order to avoid that, we have to avoid taking the broad spectrum antibiotics, and we have to maintain healthy diet, eating healthy vegetables and fruits. And we have to intake psychobiotics. It's a live bacterium like lactobacillus and bifidobacterium, which can promote our health. So it is a quotation by Hippocrates: "Death sits in the mole." Conclusion: the shift in the gut microbiota composition and stress causes the depression. Healthy individual may also experience depression due to the gut dysbiosis. This can be reduced by intake of probiotic bacteria and a healthy lifestyle. Thank you.
Next, Karthik Pandian.
Yes, ma'am.
Start your presentation.
Oh, okay. Can I start, ma'am?
Yes, you can. But be quick with your presentation. Give the essence of your slides.
Okay. Good afternoon, everyone. Let me start with my self-introduction. This is Karthik Pandian, first year MSc Microbiology from VHNSN College. Today, I am going to present food spoilage by yeast. So let's start with the introduction. Food spoilage. Food spoilage is the process where a food product becomes unsuitable to ingest by the consumer. The causes of such a process is due to many outside factors as a side effect of the type of product it is, as well as how the product is packaged and stored. If the food was spoiled by the microbe, the original value and its flavor become damaged. General characteristic of yeast. Yeast are eukaryotic cell, single cell microorganism classified as members of the fungus kingdom. Its size 3 to 4 micrometer. It was reproduced asexually by mitosis and usually show budding. Budding yeast refers as true yeast. The optimum temperature of yeast between 25 degrees centigrade to 35 degrees centigrade. The pH range between 4.5 to 6.6. Yeast in food spoilage. Yeast can be found in wide variety of environments such as soil, water, and insects. Yeast can utilize and a variety of substrates such as pectins and other carbohydrates, proteins, and lipids. Moreover, yeast are relatively tolerant to low pH and low water activity, low temperature, and the presence of preservatives. Spoilage in fruits. Fruits supply minerals, vitamins, sugars, organic acid, and dietary fiber. It's are used to make soft drinks, pickles, and sauce, and etc. Fruits are nutrient-rich substrate, but the pH of fruit does not favor for bacterial growth. As a result, yeast and molds are important than bacteria in the spoilage of fruits. The pH range between 3 to 5. The several genera of yeast can be found on fruits. Spoilage in dairy product. Yeast can grow well at the low pH of cultured product such as in buttermilk and sour cream, and it can produce of flavors described as fermented or yeasty. Yeast are a major cause of spoilage of yogurt and fermented milk, in which the low pH provides a selective environment for their growth. Common contaminating yeast of Candida species and Pichia species. Spoilage in bakery product. The most widely recognized role of yeast in bakery is a leaning agent in breads and other fermented goods such as crumbs. In many bakery products, the water activity is the most important single factor affecting the type and rate of the spoilage. There are broadly two types of yeast spoilage of bakery products and ingredients: visible yeast spoilage, fermentative yeast spoilage. Visible yeast spoilage. Many yeast can produce visible growth on the surface of bakery products, such as breads and etc. The optimum temperature for visible spoilage yeast is 15 degrees centigrade to 35 degrees centigrade, and the optimum pH about 4.5 to 4.8. Fermentative yeast spoilage. Fermentative yeast spoilage can occur in wide range of bakery material, but most common in those with high sugar content and low water activity. These include jams, marshmallow, or buttercreams used as fillings. Preservation of food spoilage. Freezing. Food kept in refrigerator, it remains fresh for some days. Germs do not grow easily in cool place. We preserve the food item such as milk, fruit, vegetables, and cooked food by keeping them in a refrigerator. Boiling. In this method, we can preserve food for a short period of time. Germs in milk are killed by pasteurization. It is for example of boiling. Salting. We can add salt to preserve pickles and fish. Sweetening. Excess sugar in food also acts as a preservative. We store food for a long time in the forms of jams, jellies, and by adding sugar. Canning. In this method, air is removed from food and put in airtight cans so that germs do not grow on them. Vegetables, seafood, dairy product are the preservative through this method. Advantage of food preservation. Germs do not grow easily in preserved food and make it safe to eat. Preservation enables us to enjoy seasonal fruits like strawberries and mangoes even during the off-season. Disadvantage of food preservation. Excess salt and sugar are used in the preservation of food, which is not good for health. Some methods of food preservation may lead to loss of nutrients. Thank you. Thank you, ma'am.
Thank you. Next is Renuka Sarojini.
Ma'am.
Start your presentation now.
Okay, ma'am. Ma'am, am I visible? Ma'am?
No? No? Present your slides again.
Okay, ma'am, I'm not again presenting my slides again. Ma'am, I'm visible now.
Yes, visible, Renivas Rajani.
Okay, sir. Good afternoon, everyone. So let me introduce myself first. I'm Renuka Sarojini I'm a third UG student from VHNSN College. I'm here to discuss about the topic study of probiotics and their isolation. Okay, probiotics. Probiotics. As a science students, surely we will hear this word at least one time, and some of us may know about this probiotics. Probiotics are nothing but which is a live bacteria and yeast which are good for our body and our health. Usually we thought germs that usually we think of that the germs will cause diseases, but probiotics are a good bacteria which is good and helpful bacteria to our body and especially for gut. Here I'm going to discuss to you about some common probiotics, and the first one is Lactococcus lactis. I have attached the image of Lactococcus lactis at the background of the slide. Lactococcus lactis is a gram-positive bacteria which extensively used in the production of buttermilk, cheese, pickled vegetables, and the manufacture of beer and wine. You know the fact that Lactococcus lactis is the first genetically modified organism used for the human disease. And also Lactococcus lactis is also a non-sporulating bacteria. And here I have attached the domain, kingdom, phylum, class, order, family, genus, and species of this bacteria. The second one I have chosen to discuss for you is Saccharomyces cerevisiae. It is a species of yeast. It is generally used in the manufacture of winemaking, brewing, and baking. Brewing is nothing but the process and activity of producing beer. The process employed here in a production of these products is top fermenting. Top fermenting is nothing but it is the process by which all type beers are fermented, and it is fermented relatively using short period at a high temperature and with the yeast rising at the top. And during the fermentation, hydroxide generated here is used as a leaving agent in bread and baking goods. Leaving agent. Leaving agent is nothing but it is a duct to make it rise used in such as yeast or baking powder. Here, the kingdom of Saccharomyces is a fungi, and division Ascomyta, class Saccharomyces, order Sarcomycetales, family Sarcomycetacea, genus Sarcomyces, species and Sarcomyces cerevisiae. And the next bacterium is Bifidobacterium. Bifidobacterium is also a gram-positive one, and it is a non-motile bacteria. It usually grows. It was usually a branched one with anaerobic bacteria, and this is a ubiquitous. Ubiquitous means that it will present everywhere, everywhere in our gut, especially in our gut. It is an M symbiotic. It uses a host. It uses a human body as a host to live its life. So we are saying it as an endosymbiotic, and it also and it mainly it was found in the gastrointestinal tract, vagina, and mouth of human and mammals, including humans. Um, Different species and strains of bifidobacteria can extend a range, which gives a beneficial health effects, and it also include the regulation of intestinal microbes in the form of homeostasis. Here, bifidobacterium is especially known as a gut microbes, which can prevent diarrhea and constipation and also reduce serum cholesterol. It also helps in improve lactose intolerance condition, also anti-tumor. It can help in the synthesis of vitamins like B6, B2, and B12. It also suppresses the growth of pathogens. Here, and the next one is isolation of probiotics. So we are going to discuss about what are the steps of isolating the probiotics in a general way. It has namely 5 steps: sample collection, preliminary screening, isolation of bacteria, identification of isolates, determination of microbial activity. And here the first step is sample collection. First, it is the step of collecting diary samples from different regions and storing it at 4 degrees Celsius, which means a low refrigerated temperature to avoid contamination. And the second step is preliminary screening. We are performing the preliminary screening with the help of phosphate buffer solution at a pH 3, and it should be performed for 3 hours. And the third step is isolation of bacteria. Here we are isolating the collected bacteria dairy products using MRS medium. MRS medium is nothing but a D-man Rogoso Shaper agar. After dissolving this using MRS broth, we need to shake that broth homogeneously, and we are incubating it at 37 degrees Celsius for 24 hours in aerobic condition. And the fourth step is identification of isolates. After incubating, the bacteria has been going through the biochemical and morphological test as a growth test at 4-degree and 5-degree test tubes containing MRS broth and the fermentation of carbohydrates. And the final one is determination of antimicrobial activity. The antimicrobial activity. Some of the acid and bile resistance can be isolated separately using the antimicrobial activity, where the pathogenic bacteria were isolated using the well diffusion method. Here, the benefits of probiotics. Generally, we have seen many use of probiotics, but here I have been listing out some few more points. Balancing friendly gut bacteria is the best example for this balancing friendly gut bacteria is Bifidobacterium, which cause no effect to our human body. Also, it improves our mental health. And the third step is it can prevent and treat diarrhea. Probiotics. Also, it can help us in easing our constipation process. Also, it reduces symptoms of digestive disorders and arming immune system. Also, probiotics helps in the field of men's health and ulcer prevention. In women's also, it plays a major role in preventing women's health and UTIs, which means urinary tract inflammation. Also, it helps in it helps it helps in probiotics also give a greater prevention in kids health and eczema, weight management, also heart health and lower cholesterol levels. Now we are going to see the disadvantage. However, whatever the topics or methods we are using, there will be a disadvantage. Same as a probiotic also has some disadvantage. Such a disadvantage is taking larger amount of probiotics may cause digestive issues like gas and bloating, and the Also, it causes severe diseases like lactobacillus septicemia. Also, it also discomfort the gastrointestinal tract and it may causes allergy. Thank you. Ma'am.
Thank you. Thank you, Ms. Renuka.
Thank you, ma'am. Thank you, ma'am.
And we like to express our heartfelt thanks to Dr. Anantaraj to act as a chairperson for this session, sir. Our special thanks to you, sir.
Thank you. Welcome, ma'am. Please.
Now we have reached the end of the session. The admiration of genius make one aspire for excellence in life that creates the harvest so abundant. Now let me take this opportunity to invite Dr. M. Kannan, Assistant Professor of Zoology, to deliver valedictory address.
Good afternoon to all. Dear Chief Guest, Chairpersons, participants, and my dear organizing members of this great event, once again, good afternoon to all. I am very much happy to share my views during this international virtual seminar on neutrinogenomics valedictory address. Before my session, that I wish to convey my heartfelt regards to the Managing Board of our VHNSN College and our beloved Principal, Dr. P. Sundarabandyan sir, and our self-financing coordinator, Dr. R. Palaniappan sir. Dear students, nowadays the maintenance of health and the prevention, treatment of chronic disease are influenced by naturally occurring chemicals presenting in the foods. In addition to supplying the substrates for producing energy, a large number of dietary chemicals have a bioactive that they alter the regulation of our biological process, and either directly or indirectly the expression of genetic information. Nutrients and bioactives may produce different physiological phenotypes among individuals because of genetic variability, and not only alter the health but also disease initiation, progression, and severity. Nowadays, the study and application of gene-nutrient interaction is called nutritional genomics or NUTRIGENOMICS. The NUTRIGENOMICS concepts, research strategies, and clinical implantation are similar to overlap those of pharmacogenomics. And both are fundamental to treat the disease and maintenance of optimal health. Nutrigenomics is a science studying the relationship between human genome, nutrition, and health. Hence, the nutrigenomics corresponds to the use of biochemistry, physiology, nutrition, genomics, proteinomics, metabolomics, transcriptomics, and epigenomics to seek and explain the existing reciprocal interaction between genes and nutrients at a molecular level. People in the field work towards developing an understanding of how the whole body responds to a food via system biology, as well as single gene, single food compound relationship. In addition to the effect of genes on the phenotype, gene can also represent response to the environmental influences, of which nutrition is one such influence. Some key nutrients of note include those involved in the carbon cycle, such as folate and vitamin B2, B6, and B12, and other vitamins such as vitamin A, which is regulate gene expression. Although progress is being made in each of the individual omics field, effective integration is required in order to provide more comprehensive phenotypic profile. Recent editorial in Genes and Nutrition by NUGO emphasized the importance of taking a system approach in future research. With human research studies that incorporate the totality of diet interaction required in order for nutrigenomics to reach its full potential. In bringing together the science of bioinformatics, nutrition, epidemiology, molecular biology, and genomics, much remains to be both discovered and determined, but future nutrigenomic research will no doubt provide further introducing insight into both nutritional science and the human genomics. My dear students, once again, thank you for giving this opportunity to share my ideas in this great occasion. Thank you.
Thank you very much, sir.
Thank you.
Champions are made when potential are converted to performance. Work hard in silence and let the success be our noise. Now, I humbly request our principal, Captain Dr. P. Sundrapandian, sir, to announce the best oral presentations.
Good evening to one and all. At the outset, I'd like to congratulate the Department of Microbiology and BWOC, Food Safety and Quality Management, for successfully organized this wonderful international virtual seminar. And I also thank all the resource persons and participants for their wonderful participation in this program. I'm really happy to declare the best oral presentation IVSN 2021. The best oral presentation, first prize goes to Jaxi Sunil Radhakrishnan, Seva Sadan RK Talreja College, Thane, Maharashtra, India. Toxicity study of biotreated dieffluent. I congratulate the author for his wonderful presentation in this IVSN 2021. And the best oral presentation, second prize goes to Dr. Aravind Sa, Department of Food and Nutrition, University of Calcutta, West Bengal, India. Sickle cell anemia, time to spread awareness. I congratulate congratulations, Dr. Aravind Sa, and I congratulate both of the winners for their wonderful work and oral presentation in the IVSN 2021. I also congratulate all the participants for their enthusiastic participation in our IVSN 2021. Best wishes, and I congratulate all of you. Thank you.
Thank you very much, sir. Next, I would like to welcome Dr. R. Palaniyappan, SF Coordinator, to declare the best poster presentations.
A very good evening to all of you. Respected Principal of this Temple of Learning, Dr. P. Sundrapandian, the former Coordinator of Microbiology Department, Dr. Kannan, the Head of the Department of Microbiology and BWOC, the staff members, respected participants and resource persons from various places. Really a happy occasion and an important aspect in our college, international virtual seminar on nutrigenomics, and really a wonderful situation in which several participants all over from India has participated, which shows the repute that the college is undergoing in the current scenario. It has been placed in the 53rd rank in NIRF ranking. Really, we are very much proud to say that, and we have been awarded with the DST fellowships and scholarships amount. And I'm really happy to announce the best poster presentation of International Virtual Seminar on Nutrigenomics. The title of the paper is Anti-Hypertensive Peptides Derived from Fermented Camel Milk by Proteolytic Lactobacillia in Silico in vitro study. And the author is Subrata Hatti. Subrata Hatti, congratulations, is from the Department of Dairy Microbiology, SMC College of Dairy Science, Agricultural University, Gujarat, India. And empty number of participants participated in this virtual seminar, and many number of papers have been presented. And I congratulate the Department of Microbiology, BIOC, Food Safety and Quality Management, for to have done a wonderful task in conducting this seminar and to have brought VHNSN to the limelight of our nation. I congratulate all the participants of this workshop. I congratulate all the resource person for to spare their wonderful times for our college. I thank one and all and our college management and our principal and the Department of Microbiology for to have given me this opportunity. Thank you.
Thank you very much, sir. Yes. The participants, you will be receiving e-certificates for your best presentations. Now it's time to wave a goodbye 1,000 times to the cheerful and wonderful participants with a side of pride and gratitude from our side. Now I request Dr. T. Shanmugaprabha, Assistant Professor of our department, to propose vote of thanks. I welcome you, ma'am.
Good evening to Anandal gathered here. On this wonderful evening, I would like to present the vote of thanks. We thank these golden opportunities to thank the respected chairperson of this virtual seminar on nutrigenomics, Dr. V. Arvindraj sir, for attending this function in his busy schedule. We will cultivate the advice you gave us in real life. Thank you, sir. I extend my gratitude to the respected principal, Captain Dr. P. Sundarabandyan sir, not only for spending his time with us, but also for encouraging us to achieve many endeavors. Thank you, sir. I extend my gratitude to the respected SF coordinator, Dr. R. Palaniappan, who is our mentor in helping us to advise our milestone. Achieve our milestone. Thank you, sir. It's my pleasure to thank Dr. M. Kannan, Department of Zoology, for guiding us for the virtual seminar. Thank you, sir. I heartily thank Dr. Prem Sangar sir and Dr. P. Govindarajan sir for their valuable support. I thank our participants from various state and college. Last but not the least, I thank my colleague and my fellow students for enthusiastically arranging this function. Without you, this function would not be a success. Thank you, buddies. I sincerely thank all the non-teaching staff and the technical members for glorifying this function. Once again, I thank you one and all wholeheartedly. Thank you.
Thank you very much, ma'am. Now let us conclude the session with the national anthem. the feedback link. We request the participants to fill the feedback link to have your e-certificates. The best poster and oral presentation, for best oral and poster presentation, you also be receiving e-certificates within a week.